ஹலோ. இல்ல வேற யாரும் பேசலையே.
Yes. Vivek.
हेलो
टीम वाली स्क्रीन देख रहा ना तो चल रहा है।
हेलो
Yes, ma'am.
doctor, can you please, you're inside, have you entered? Okay, we were waiting for our NC to enter because there was a problem. Yes. Now let me see whether she has entered.
Yes, ma'am.
Avmc, Dr. Kalaimani. Okay, thank you.
Yes, ma'am.
Pankaj ji, make me co-host also.
Okay, maybe. Okay. Okay. चलिए
सर मेरे सेकंड डिवाइस को कोहोस्ट बना दीजिए, दोनों डिवाइस को कोहोस्ट ही रखिए।
Biomedical science conference.
பிஎம்சி கான்னு ஒண்ணு இருக்குல்ல, அதுல ஒரு சேரிங் போட்டிருக்காங்க. என்னவோ வருது, நமக்கு பாதி புரியவே மாட்டேங்குது. வர வர கனெக்ட் ஆயிடுச்சு. நேத்துக்கு தெரிஞ்சிருந்தா வேணான்னு இருப்பேன். ராஜன் ஃபோன் பண்ணி ஓப்பன் ஆக மாட்டேங்குது வீடியோ.
Hello. Good morning, Deepti ma'am.
हाँ खाली छोड़ी है
Good morning to one and all. It gives me immense pleasure and pride to welcome you to the 6th International E-Conference, BMS Econ 2026, organized by the Departments of Anatomy, Physiology and Biochemistry, Arubadai Veedu Medical College and Hospital under Vinayaga Missions Research Foundation. This year's theme, Beyond Microscope, Transformative and Future Strategies in Basic Medical Sciences, beautifully reflects the evolving landscape of medical education, research and innovation. I am Dr. Kalaivani K., Assistant Professor, Department of Anatomy, and I feel honored to be your Master of Ceremonies for BMS Econ 2026. Without any delay, let's move on to the first scientific session of the day one, BMS Econ 2026. Our first session sets the tone for this conference, focusing on the integration of artificial intelligence in basic medical sciences. Respected dignitaries, esteemed faculty, distinguished delegates, and dear participants, it is my privilege and honor to introduce the chairpersons for this scientific session. Dr. Deepti Shastri, ma'am, Dean Faculty of Medicine, Vinayaka Missions Research Foundation, Deputy Dean, Vinayaka Missions, Kiripananda Warrior Medical College and Hospitals, Salem, Tamil Nadu. Ma'am has 28 years of experience
You are mute. Dr. Kalevani, you are on mute. No, we couldn't hear you.
I would like to introduce our next chairperson, Dr. Kalpana Ramchandran ma'am. Ma'am is professor and head Department of Anatomy, Sri Ramchandra Medical College and Research Institute, Chennai. She is also National Coordinator, NPTEL DTH Medicine, and co-convener ACME, SRMC, NMC Nodal Center for Faculty Development. Ma'am has completed MS Anatomy and DNB Anatomy in 2001, and ma'am was awarded Famer Fellowship in Medical Education 2014 and ACME in 2017. And ma'am has conferred FRCS by the Royal College of Physicians and Surgeons, UK in 2020. Ma'am has won many awards, including Dr. M. Cooper Memorial Award twice and Best Teaching Award. Thank you. I request the chairpersons now to commence the session.
Thank you for the introduction, Dr. Kalaivani. Good morning, everyone. This is Dr. Deepti Shastri. At the outset, I would like to congratulate the organizing team of BMS Econ 26 for organizing this event every year, undeterred, come COVID or elections. I would like to thank Dr. T. Rajan for giving me an opportunity to chair this session. It is a great honor to welcome Dr. Parthiban Srinivasan, an erudite speaker, to open the innings of the 6th International Conference of BMS Econ 26. His expertise on how AI can change basic medical research would give us fresh insights into how translating computational ideas into real world experiments would benefit us, whether it is data fairness or trust issues or building collaborations for future breakthroughs. I would like to invite Dr. R. Kalpana, the co-chairperson, to introduce our erudite speaker.
Thank you, ma'am. Am I audible?
Yes, Dr. Kalpana.
Thank you, madam. Good morning, everyone. I thank Professor Rajan for the opportunity given to chair this session. And it's a great pleasure to introduce the speaker of this session, Professor Dr. Parthiban Srinivasan. Professor Dr. Parthiban Srinivasan is an experienced data scientist. He earned his PhD from Indian Institute of Science, specializing in computational chemistry. Sir, after his PhD, continued research at NASA Ames Research Center, USA, and Weizmann Institute of Science, Israel. Sir then worked at AstraZeneca in the area of computer-aided drug design for tuberculosis. Later, Professor Parthiban Sir headed informatics business units in Jubilant Biosys and then in GVK Bio before he floated the company Parthis Reverse Informatics and later on an AI consultancy, Vignani. Sir then returned to academia as a professor of data science at the Indian Institute of Science, Education and Research, Bhopal. Professor Parthiban is currently professor and director at the Center for AI in Medicine, Arbade Vidu Medical College and Hospital, Vinayaka Mission Research Foundation, Puducherry, India. Today, Dr. Parthiban Sir will be speaking on Beyond the Microscope, AI and the Future of Medical Discovery. This topic is highly relevant and exciting topic, and I'm sure we are all going to benefit from your valuable insights, sir. We will please join me in welcoming Professor Parthiban, sir. The floor is yours, sir.
Oh, thank you very much. Thank you very much, Dr. Kalpana, for the kind introduction. And also thank you, Dr. Deepti. And so before I start, can I share my screen? Will I be? Yes, sir. Please share. Just share the screen. Can you see my screen? Are you?
Yes,
Are you
sir.
able? Okay, so I'll go to the full slide mode. Are we all set now?
Yes, sir.
If you can, okay, I can start now, right? My time starts, right? Just say yes, I'll start.
Yes, sir.
Okay, thank you so much. So, good morning, everyone. It's a pleasure to be here and to be part of this conference on such an important theme. Today, I would like to talk about something very simple but very powerful. How do we move from seeing to understanding? So, that's the idea behind my talk. Beyond the microscope, AI and the future of medical discovery. And my name is Srinivasan Parthiban, and heading the Center for AI in Medicine at Arubadai Video Medical College and Hospital, VMR of Puducherry. And by the way, today I am joining from San Diego in US. So at the outset, I would like to thank Professor Rajan and all the organizing committee members for giving me this opportunity. And it's truly a privilege to be the opening speaker for this conference. So let me little move this. Yeah, okay. So for decades, medicine has been built on what we can see. The microscope has been our window into disease. But today, we are beginning to realize that seeing is not enough. And we kept going deeper from the whole body to organs, to cells, and to molecules. At every stage, we believed that if we could see more, we could understand more. But the real question is this slide not moving? Oh, but the real question is, have we actually understood disease? So that's the question. So if we go back in time, before the invention of the microscope, medicine looked very different across different traditions, whether it was Ayurveda or Chinese medicine or early Western medicine, the approach was largely the same. We observed the human body from the outside. And then everything changed. The invention of the microscope allowed us for the first time to look inside the human body because no longer it's invisible, because we could see cells, we could see the structures, and eventually we could see the genes. So medicine kept going deeper and deeper, but something unexpected happened. The more we saw, the more complex disease became, from anatomy to cells to genes to massive amounts of data. So we moved from clarity to overwhelming complexity. And that brings us to how we diagnose disease today. We began by looking at the whole body. Then came like the stethoscope and imaging techniques like X-rays, followed by laboratory tests, and later we moved into the MRI, and then we went even deeper from organs to cells, and eventually to the molecular level disease. And now we are beginning to see artificial intelligence to perform many of these same tasks. But even with all this, something is still missing. Let's go. So we are limited by what we can see, and we are limited by what we can understand. So let me repeat. We are limited by what we can see, and we are limited by what we can understand. So that is where the real challenge lies. So meaning today, we can see more than ever before. So we can see organs, we can see cells, we can see molecules, signals, and entire biological systems. But the problem is not visibility, it is the complexity. And we are surrounded by data, imaging data, genomics data, clinical records, but we do not always understand what it means. So what it means is we do not understand it. And to do that, we need different way of thinking, because seeing is not enough. So we can collect more data, generate more images, and monitor continuously, but still understanding does not automatically follow. So, meaning we need to move from observation to interpretation. So, the question is, how do we move from seeing to understanding? So, that is the question. So, meaning, uh, so the how do we move from seeing to understanding? The answer is artificial intelligence. So AI allows us to take large amounts of data from images and find patterns. And that's all, I mean, that's not obvious to the human eyes. So it does not just help us to see more, it helps us to understand. And that is the shift that we are seeing from seeing to kind of understanding. So, but today the AI is no longer it's a buzzword, but as a way of thinking and a way of it's a complex medical data, and we started to begin to understand it. So, what do we mean by artificial intelligence in the context of medicine? So, artificial intelligence is the ability to recognize. I think this one is okay. So, artificial intelligence is the ability to recognize patterns in complex medical data and convert them into understanding. So, these patterns could be in images or physiological signals or even biochemical data. So, in simple terms, AI helps us to move from data to insight. Now, let's move on. So, and this is the beginning to transform the very foundations of medicine. So, if you look at the medicine at its core, uh, it can be understood across three fundamental domains: anatomy, which is about structure; physiology, which is about function; and biochemistry, which is about molecules. And interestingly, so AI is transforming each of this in a different way. In anatomy, we move from observation to representation, and in physiology, we move from monitoring to prediction, and in biochemistry, we move from measurement. So we move from measurement to understanding the patterns. So let me walk you through each one of this, starting with the anatomy. Here, now, let's look at the artificial intelligence in anatomy first. So here, anatomy, when we say that, I mean, you're all medical doctors, so it's not that I can teach you what is anatomy. So anatomy, I'm talking from the artificial intelligence point of view. So anatomy is fundamentally about the structure. We study the human body at different levels: organs, systems, tissues, and cells. So, and for a long time, this understanding, this one, the organs, systems, tissues, and cells, this understanding has been largely visual. We observe, we describe, and we interpret. But today, this is beginning to change with the help of artificial intelligence. We are now able to convert this structural understanding into digital representation. So what you see here is not just a human body. It is a connected representation of anatomy. So where information flows from cells to tissues, tissues to organs, organs to systems. And this is the key shift from structure to digital representation. And this forms the foundation for what we are going to see all the rest of the part of the anatomy stuff. Now let me move on here. So what we have seen so far is the anatomy, which is a structure. But with AI, this begins to evolve further. So we move from anatomy to something more dynamic, which is a digital twin. Traditional anatomy is static, whereas which captures the snapshot of the human body at a given point in time. But the digital twin, this is very different. It is a living representation of the human body, and it is continuously updated over time. So it integrates anatomy with the data and with time. So what you are seeing on the right is not just a model, it is a system that evolves and a new data comes in. And this is the key transformation from a static view of anatomy to a dynamic, continuously evolving digital twin. And this is the distinction in which anatomy is heading. This is the direction. This is the direction towards the field of anatomy, which is running. So now let's look at the digital twins in medicine. Let's narrow down from the whole anatomy into the digital twin. So they can be applied across multiple domains: oncology, cardiology, neurology, maternal fetal health, and orthopedics. So each of these areas represents a complex system where understanding the structure and the dynamics is critical. And AI is beginning to enable digital representation across all these domains. So, but today, actually, as you would have seen, there are oncology, cardiology, neurology. You would have seen, you probably might be seeing a yellow color rectangle around maternal fetal health. So today, I would like to focus on the one of these areas, the maternal fetal health, and this is uniquely challenging domain, because here where two interconnected systems, the mother and the fetus, evolve together over time. And understanding this interaction is not just important, it is critical for every detection and prevention of complications. So for the rest of the next couple of slides, we will focus on the how digital twins can be applied in maternal and fetal health. So let's now look at this in the context of maternal fetal health. Here, the idea is to build a living AI model that integrates multiple sources of information. So as you can see on the left side, you can see all the different sources of information: maternal data, fetal data, imaging, and genomics. These are not isolated data streams. They are all interconnected, and they evolve continuously over time. It's not just a static data. So the goal is not just to observe, but to predict and prevent complications before they manifest. And this is where the digital twins become more powerful. They allow us to bring together all these data sources. So what you are maternal data, fetal data, imaging, genomics, all of them put together so that it is trying to bring it in a extraction, normalization, deduplication, and integrating the whole stuff together, and continuously it is updated representation. Such a system can support clinicians by providing insights and enabling better decision making. And within this framework, and we can focus on specific clinical problems such as preeclampsia, fetal growth restrictions, what you are seeing on the right. Actually, we have put something around 20 such issues, and these are some of the higher prioritized ones. So which preeclampsia, fetal growth restriction, gestational diabetics, and preterm birth. So each of these represents a scenario where early understanding can lead to better outcomes. Now let's move on. So to truly understand a living maternal fetal digital twin, one kind of model is not enough, because what we have seen in the past is some research groups, they move with the physics-based models. So sometimes, you can define the physics of a chair, physics of a table, but then physics of a biology is not pretty well defined. So there is a risk of just restricting to physics-based models. So we are going to bring in all possible, the best of every one of them: physics-based models, foundational models, and agentic AI. So the agentic AI is currently the hottest area. When you say even AI, by default, it is agentic AI in the AI field today. So it requires the integration of multiple layers of intelligence, each addressing different aspect of the problem. So the first, what you are seeing on the left side is the physics-based models, and these help us to simulate the underlying biology, maternal circulation, placental blood flow, and the fetal growth dynamics. And they ensure that what we build is grounded in physiological reality, not just data patterns. So we are not just representing the data, statistics, probabilities. It's a pure science. So second, we need foundation models. So the foundation models meaning, so typically when you are talking about ChatGPT, which are normally coming from large language models, meaning which are all trained on the English language, the internet of all the sources of English language. But then when we say foundation model, it is multimodality, because the medical subjects is not a single modality. It is imaging, clinical records, genomics, proteomics, the drugs, which are chemistry, the longitudinal data. So this is a multimodal. So the model needs to be trained in a different modalities. So it's such a so they help move the whole system forward. And the finally, what you are seeing in the right side, which is the agentic AI systems. So these systems continuously monitor data, predict risk, reason over evolving conditions, and support clinical decisions. So the reason for agentic AI, so this agentic AI, when people are talking about AI, this started from 2024. They brought this agentic AI system, and now this is currently getting accepted in every single possible area: healthcare, medicine, drug discovery, biology, pharma, financial, and manufacturing industries. Everywhere, this agentic AI, which is proving to be pretty successful one. So when these three come together, we move beyond microscope, from images to mechanisms, from measurements to meaning, from monitoring to action. And this is what enables us to build truly living maternal fetal digital twin. So one of the reasons why I picked up as part of anatomy is that we together from AVMCH and the AV SAS in the Mahabalipuram campus, we have written a proposal on a digital twin for this maternal fetal health, and it has crossed one level. It has moved to the next level, and we have made this proposal, and this is pretty much, we are actively working in this space at this moment now. So this is just wanted to share this aspect with you. So now from here, now the second aspect, which is physiology, AI in physiology. So now let's move to this domain, physiology. Now from anatomy, that's about structures. So physiology is about body functions, and these functions generate signals over time. So, and these signals are continuously produced, for example, from the heart, the brain, and the other body systems. So today we can capture these signals in many ways through ECG, ICU monitors, and wearable devices, and so on. So unlike anatomy, which looks at structure, and physiology is dynamic, it changes over time. And this continuous flow of data creates an opportunity for artificial intelligence. By analyzing these signals and learning patterns, we can move from simply observing the data to predicting what may happen next. And this is the key, from continuous signals to clinical prediction. So let's move on. So to make this signal useful, we need to process them. So the processing is what something we call it as a feature extraction. So physiological data usually comes as a you know pretty well, it's a time series data, signals that change over time. But for building predictive models, we need to convert this into a table of data, the structured data. So this is done through the step called the feature extraction, where we can summarize the signals into meaningful values such as averages, trends, and variations. The other important step is controlling the level of detail, or the granularity. For example, we can look at the data every minute or every 5 minutes, depending on the problem. So these steps helps us to convert the raw signals into structured data that can be used for prediction. So it's like any financial data, you have an Excel sheet, each column which is talking about a particular property, or in machine learning language, we call that as a features. So this is each based on that, we can build a model. So in a simple way, if I have to tell you, if I ask you 1 plus 1, which is 2, 2 plus 2, 4, 3 plus 3, 6. This is rule-based approach. But if I tell you 1 plus 1, 11, 2 plus 2, 22, if I ask you a question 3 plus 3, you would say 33. What is happening now is, when I gave you two examples, 1 plus 1, 11, 2 plus 2, 22, you build a logic in your mind. And when I ask 3 plus 3, you apply the logic onto the question and you are giving the answer as 33. So that is precisely what we are trying to achieve here. So when I have a data for many patients, from that, humanly, it is impossible for me to build a logic. So this is where I'm trying to bring in the machine learning AI. We want the AI to build that logic. So when I have that logic, when a new patient data which comes in and without any diagnostics, then my logic will be applied onto the data and then I get the diagnostic. So that's a pretty in a simple way that I can put this in here. So from the raw signals to predictive tabular data. So now let's look at the how clinical data has evolved using the ICU as an example. So because you probably might know, many of you, the MIMIC is a data which was originally in the version 1, which is a completely a hard copy. Then it was created in a digitalized form. Then it is very well curated 3, and then the fourth version is the AI ready data. And many of the people, you take the data because this is open source and freely available. And pretty much this data comes from the ICU data, the patient's data. And so when you have the data, you can start building the model. And from that model, you can leverage those models for your own purpose. So we have signals such as ECG, blood pressure, oxygen levels, lab results, and all these measurements. And earlier, much of this data was recorded on the paper, and then later it moved on to digitized form. And in version 3, it was normalized. So this is the next stage, and the systems improved. So, and today we have AI-ready data sets. So this is the key evolution from raw patient signals in the ICU to structured AI-ready clinical data. So when you have the data, you can build a model and then you can apply your regular practice. So this is what the change which is happening, because this was the biggest bottleneck. See, building a model is pretty easy, but then having the data which is in a digestible form for AI. So that is the biggest bottleneck that we have been facing. But through many initiatives, now we are getting the data in the open source form. Now, so far we have seen how the data is generated and how it evolves in clinical settings. To build strong predictive models, we often start with the large global data sets. So I think I mean, all these medical professionals, they should know about. Sorry? They should know about UK Biobank, because UK has taken the initiative of collecting the data from 500,000 people, 5 lakh people, the human data which is available. And you just need to, there is a small formalities you just need to complete, and then you will have access for free all the entire 5 lakh patients data with you. It has a comprehensive data. So when you have that kind of a data, and when you build a model, then you will have the most powerful system in your place. So one of the reasons which I am focusing on this is, in Arubade Vidu in Pondicherry facility, we are also trying to build one of the repositories, image repositories. So we have taken that initiative and we made a proposal, and then it has also reached a couple of levels. And so we are also in an advanced stage in terms of applying for the grants. So assuming things will fall in place, then So this is one of the initiatives from India. So like already US has such similar initiatives, and UK, and then in Indian government is also taking such initiatives, and from multiple locations, and from Pondicherry we have also made this proposal and made a very good progress in this right now. So what we have seen so far is how physiological data can be used for prediction using machine learning and deep learning models. We can learn patterns from high resolution ICU data and identify early signs of deterioration. And one important example is sepsis. And sepsis is a serious condition where the body responds to infection in an extreme way, and it can progress very quickly. And if not detected early, it can lead to organ failure and even death. So, but the key point is early prediction can make a major difference. And there are like Johns Hopkins and many all the leading popular universities and the hospitals, they were trying to build the models for sepsis prediction. And in a similar way, and our students also started working on sepsis data, and then we are trying to build the predictive AI models to detect sepsis risk early. So this is one of the other work that we are currently working on. So from data to prediction, and from prediction to early intervention. So that is about the physiology. Now let's move on to the third aspect, which is AI in biochemistry. So let's look at this. Here at this level, we are looking deeper, at a deeper meaning at a molecular level inside the body. This includes proteins, genes, and other molecular components that control how the body functions. And this is where AI is playing much bigger role. And AI can be used across multiple areas in biochemistry. And what you are seeing on the right side, the biomarker discovery, then drug discovery, understanding mechanism of action, and precision medicine. And now let's focus on one important application, like sort of a biomarker identification. So biomarkers are the measurable signals that can help us detect disease early, predict risk, and of course, understand disease progression. So traditionally, finding biomarkers was slow and required a lot of manual effort. So, but then with the arrival of AI, we can analyze huge amounts of data, molecular data, and discover patterns that are not easy to see. This helps us to move from molecules to biomarkers, and then from biomarkers to clinical insight. And if you look at the second one, the drug discovery, the target identification, molecular design, see the identifying the target. So let's say that our human body has about how many proteins that we have, something around 25,000, some measures will say 30,000 or 20,000. But then the number of drugs, all the FDA approved drugs, they are not targeting anything more than 700 biological targets. So identifying the right targets is the biggest issue. So associating the target, either it's a kinase, protease, GPCR, ion channel, nuclear receptors, whatever it is. But then associating with the disease, that's where we are still finding a biggest trouble. And validating any novel new targets is not that easy. But you all know that the biomedical research in the last 20, 25 years, the whole world has invested more than 2.7 trillions of dollars, and the result of that is sitting in the form of journal articles and somewhere in the billions of pages of information. That's where the many of these solution to these problems are sitting. And it is humanly impossible for us to access every one of them. So this is where AI, which comes in, connecting the dots, makes sense out of it, finding the target for a given disease. And once I have the target, now I can, with the active site, I can build a molecule, generate the molecule. So for the AI, until a few years ago, AI means predicting. You give a molecule drug and it will predict whether it is good or bad. But now I don't want the prediction. You tell me what drug that I should synthesize. So this is the kind of a change, which is paradigm, which is happening. And in fact, there are many success stories in the last 1 year which is coming. The drug which was completely designed by AI, which is entering into the clinical trial, and it has crossed the clinical trial 2. Now it is moving into the clinical trial phase 3. So that is the success of AI that we are witnessing at this moment. This is the reason everybody is trying to get into this space. Now, as I said earlier, the kind of a lot of targets, proteins, pathways, and small molecules. And at this level, we are looking, so some more deeper. The thing is, uh, the biological pathways over here, many of these targets have been well studied. We now know all these. But the problem is the mechanism of action. We know this is the target and this is the disease. Both of them are associated. And we have been thinking that this is the mechanism of action. But then for many of these drugs and the proteins, the mechanism of action that what we are assuming, still it is not concretely, it has been established. And even if you take the latest research article, that will talk something different. So the thing is, we need to have a repository of mechanism of action, how these drugs which are interacting in the human body. So unless we understand clearly, we cannot make any improvements further. So this is where AI now, it is playing a much bigger role. What it means is that, let me take to the next, let me explain. The chemical probe database, which has about 1,200 curated compounds, that is the drug interacting with the protein for a given disease. What is the mechanism of action? What is the total knowledge that we know about that specific mechanism of action, which is in a granulated AI digestible format, in an Excel sheet format, which is sitting inside. 1,200. But then how many data points would be there, you think, in the literature? Millions of data points. But so far, manually, we could be able to only collect 1,200. And this is actually, in fact, I'm in San Diego. Yesterday, there was a talk from Stanford University. Stanford University, they brought this problem to Google. And in Google, that technology professionals and Stanford University doctors together, they cracked this problem. Meaning the mechanism of action. I've been keep talking, so you can give me the signal so I can stop whenever, or 5 minutes before you tell me. So the Stanford University and Google, they have cracked this issue. Now within few weeks of time, now the number of the chemical probe database is 10 times it has been populated. And then there were some kind of a 5% error. The error is essentially when you compare AI has curated certain mechanism of action, which is not in alignment with the 1,200, 5% of them. When they manually analyzed the AI result and the manual one was outdated. The information sitting in the database was from the publication in year 2026, whereas the AI, it is able to pull out some data from which was published in 2023. So what I mean is the database, that manual effort that is going is static, whereas AI, it continuously updating this information. So this is the power which is coming here. And the biomedical field, which is doubling every 73 days. Like 10 years ago, it used to take 10 years to double. But today, the pace with which it is growing, it's humanly impossible for us to follow what is happening in your own specialization. So this is where AI is coming and helping every one of us sort of. So now, the foundation model. So everybody's talking about ChatGPT. So processing, originally the transformer, when they were proposed in year 2017 by Google, it was basically for language translation. If it is English, when this is processed by AI, it will give you German. If it is English to French or Japanese to English, that was the whole entire AI framework was built mainly for that. But then it is so surprising. It is, if you consider the drug, if you take the drug is a chemistry. Chemistry by itself is a language. So the transformer, which was originally designed for language translation, now able to understand the language of chemistry, understand the language of genomics, understand the language of proteomics. So this can connect, build the relationship which was not so obvious to the human. So that is what the foundation model is about. It is foundation model which can process multimodality, and then it is able to process the information, identify the relationship among the various stuff. So now one of the reasons why I brought this aspect is in Avmch, we have our microbiology department, we have about 30,000 images. And on this also, we made a proposal to the government that we want to build the India's first foundation model on microbiology images. This is for urinary tract infection. So we have made a proposal to the government, and this also has advanced to the next level. And so we are making some good progress here. And so that's the stuff which is related to this. So I'm happy to share that information here. So now from here, let's move on. So it's sort of, I wanted to, so I want to take a step back and look at what we have seen in this section. So we started with the molecules, proteins, genes, and biological pathways. And we saw that even though we know many targets, understanding how they actually work is still a challenge. We also saw the knowledge exist, but it is scattered across a large amount of scientific literature. And this is where AI helps us by bringing together text, data, and images and turning them into meaningful understanding. So the shift is going from molecules to biomarkers, from data to understanding, and from understanding to clinical insight. And this is what it means to go beyond the microscope, not just seeing the molecules, but truly understanding how they behave and how they impact disease. So that's what I wanted to communicate. So now I just wanted to give a little bit of from medicine. I just wanted to take a little one slide away from that. So the chat GPT, everybody, all of you are familiar. So which is actually part of a generative AI. So you are asking a question to generative AI, you are asking a question to chat GPT, and it is responding. So it is understanding your question and it is generating answers. That's all generative AI. But the world has moved. We are in agentic AI. It understands, it generates, and it acts. So now when you are talking about AI, we are talking about agentic AI. So now this is, I don't want to, given the time, I think I'm also keep an eye on this. So can I take some 5 minutes or something, or is it okay?
Yes,
How
sir.
much time do I have? Okay,
Yes,
great.
sir.
So that, okay, fine. Then let me kind of organize my stuff for 5 minutes. So this is just to give you the currently the two fields which are at the peak. One is AI agents, as you can see in the top, and AI ready data. So meaning when people say I'm working in AI, 80% of the time they are firefighting with the data. So even actually the startup companies from Bangalore, they are all now they thought that building an AI based companies they can make money out of it, but then everybody is struggling because the data, the gold mine which is sitting in the hospital, that is in Avmc, we have the gold mine. So it's time that the medical hospitals, they should start building the model. So if you look at until last year, all these AI efforts by the startup companies, 95% of them, they failed. Reason being, the technologists, they assume something and then they build a model and then come to our hospital and then they're telling that we have built a application for healthcare. And no doctors wants to believe it. No trust factor in this. So it should be the other way. It should be the domain expert. It should be a problem first approach. I have this problem within my biomarkers, the microbiology or anatomy. Okay, so AI can do this, let me leverage it. So this is called the problem first approach. So that's where the AI ready data, which hospitals, we do have it. And if they know, they will come and sit in front, I mean, in your office. So that's the stuff we need to understand the power of what we have been. In fact, for the the maternal fetal data, when I was talking to Dr. Deepa, she already says that every year she has about 600 data points. Those are all the gold mine. So we already have what is needed for building such a world-class model. So we need to revisit what are the data points that we have, how we can consolidate. So the reason why I'm saying is, for example, the agentic AI, which I have been talking about, and you also would have read the news that in China, they have built a digital hospital, AI-powered hospital. And in fact, they have 42 AI doctors. They are all agentic doctors, agentic AI-based doctors, and 4 AI nurses. Now this is in operation right now. So to understand what is that agentic AI, this is no different from the James Bond, meaning the James Bond normally typically if you watch the movie, he works on a particular mission, but then how that mission he is operating intelligently in a complex environment, he takes the decision. It's not that the mission director will tell him step by step what to do, and he'll be given the tool, and he will have the entire log history, so the mission logs, and there will be a command center. If you map it equivalently, the agentic AI healthcare, all that James Bond, you can map it like a clinical objective, which is the mission briefing, clinical decision agent, and the Q gadgets that James Bond who is using, that is nothing but electronic health records, imaging tools, and so on. And then medical knowledge retrieval, patient history, care coordination system. So this is what currently the hot area, and then this is already started to deliver the results. Earlier, few years ago, AI promised a lot, but miserably failed to deliver what they have promised. But then right now, there is so much of hype around going on in the agentic AI, but at the same time, many of these projects are getting benefited out of it. So that's the reason everybody is getting encouraged. So for instance, so I'll take a couple of minutes. Okay. See, only the 2 points. So for example, if a patient who is affected by, let's say, that stroke, it's not that one single doctor is enough to take care of that patient. You need a cardiologist, whether his heart is functioning well, and is he a diabetic patient. So everything after that, the neurosurgeon comes and do the operation. It's like a coordination. So for here, until year 2025, people have been talking about single agent. Now there'll be an agent specialized in neurosurgery. There'll be an AI agent specialized in cardiology. There'll be a specialized agentic AI for diabetics and so on. So it's like a music, the orchestration, the medical orchestration, which is happening. This is what we are trying to build. This is where the world is moving towards. I just wanted to tell this part. So the expectation is AI will save 10 million lives by 2040. And let me move on here. So these are all the normal one. Let me quickly wind up. So now this is the important slide. So let me stop with this. So now the slide shows how new technologies have shaped medicine over time. When imaging technologies were introduced, they led to the creation of radiology. When molecular assays became available, they gave rise to the clinical genetics. And when tissue microscopy became widely used, it shaped modern pathology. So each major technological shift, it's not just improved the medicine, it created entirely a new specialty. And today, we are seeing a similar shift with artificial intelligence. So, meaning AI is not just another tool, it is becoming a core part of how we understand disease and make decisions and deliver care. So, which means AI now requires the same level of professionalism as radiology, genetics, and pathology. This includes training, standards, validation, and clinical integration. So, what we are witnessing today is not just the adoption of a new technology, but the emergence of a new discipline in medicine. So, with that, the bottom line, the one take home message is the microscope helped us to see disease. AI will help us understand it, and understanding is what saves lives. Thank you. I'm done.
Thank you. Thank you so much, sir, for enlightening us on the vast opportunities that AI provides for basic medical research as well as clinical studies. And I would like to congratulate you on working on developing digital twin as well as the microbiology UTI data repository. I hope you experience success in both the fields.
Thank you so much, ma'am.
Yes, sir.
Yes.
The session is open for any questions that the audience has. Sir, by the time audience questions are posted on the chat box, I have a question of for myself. You
Sure.
mentioned about Dr. Deepa as well as the hospital being a gold mine for
Yeah.
providing data to create
Mhm.
AI models. So I would like to know how can we arrange or encourage sharing of this diverse, high quality data while protecting the privacy of these institutions and also following the local laws because it's open not only for our country but globally AI models. So how do we protect the privacy of our own patient data?
Oh, yeah, that's a very interesting topic. In fact, this is something almost, it's not a matured topic yet because this is the reason almost policymakers, people, everybody, they are coming for the privacy, policy, and protection. So these are the three major aspects which so far we have not given due attention to all this, and now it is almost every hospital, every organization, including government, is taking initiatives. And in fact, we are also planning to have around end of April, Sam has planned something related to this in Pondicherry, having bringing all the experts, having a panel discussions on the data, legal, policy, and cybersecurity on this. Because another thing is the federated learning, meaning let's say that the hospital A is competing with in the business with the hospital B. But at the same time, hospital A has 10,000 data, hospital B has 10,000 data. If both of them, if they share the data, each hospital will be benefited. So the way the things are going is a cooperative competition. Something new terminologies are coming. That is, in Arubadai Video, if I build a model, my model will be trained on my data, but also the model will have a connection to the hospital, my competitor's data. And the competitor data will not be shared with me, and vice versa. When I have a so these are all the new things which are emerging. It's a little early now, but then we are all aware of it. So it's too early to give a very concrete answer for these questions, ma'am.
Thank you, sir. Similarly, I guess we need some systems to review the claims by AI before we pick drug targets, as you mentioned, or clinical studies. Because when we do real world experiments, we always validate the data. And you mentioned the same now. Validation and standardization are required for AI patterns as well. So we need to have systems which actually review these AI claims.
In
I
fact, the
guess.
year 2026 belongs to validation, ma'am. 2020, everybody, wherever we go, immediately they say, where is the validation? Because the agentic AI that we are talking about, there is no validation at all or anywhere. So year 2026, everybody is talking about validation. So you can claim anything, but unless there is a validation, especially the medicine, there are two extremes. The technology people, they always get fascinated by the real world problem what we have. The real world problem, the medical hospital, we are always skeptical about whenever the technology comes in. So unless we get thoroughly convinced, we will not adopt completely. So that is there. It's a two end of the spectrum. So the extreme end of the spectrum that we are talking about. So that's where we need to bring in the balance.
Yes, sir. Thank you so much once again for your enlightening talk. I'm sure all the participants have benefited.
Okay. Thank you, ma'am. Thanks a lot.
Thank you, sir.
Thank you, sir, for that insightful and thought-provoking session. Thank you, ma'am, Dipti ma'am and Dr. Kalpana ma'am, for chairing the session. On behalf of organizing team, we would like to felicitate the speaker with e-certificate and e-memento. Now we proceed to felicitate our chairpersons, Dr. Deepthi ma'am.
Thank you.
And Dr. Kalpana ma'am.
Thank you very much.
Thank you, ma'am. Thank you, sir. I now request all the dignitaries and resource person to switch on their cameras for a group photo.
Not studying that. Huh? Why 6, sir?
Please switch
Now
on
you are
your
please take
cameras
that.
for a group photo,
Sixth
ma'am.
semester.
Sir.
Introduction is over. Huh? Tell me the progress of your thesis.
Dear madam, please mute, ma'am. Dr. Kalavani, you can proceed.
Yes, sir. Yes, sir. Respected dignitaries, esteemed faculty, distinguished guests, and dear delegates, a very warm and pleasant morning, everyone. I welcome you all to the inaugural ceremony of 6th International Conference, BMS Econ 2026, organized by the Departments of Anatomy, Physiology, and Biochemistry, Arubadi Veedu Medical College and Hospitals, under Vinayaka Missions Research Foundation, deemed to be university. To begin this auspicious occasion, let us invoke the blessings of the Almighty. I request all the dignitaries to kindly join for the invocation song, followed by the ceremonial lighting of the lamp, symbolizing the dispelling of ignorance and ushering in of knowledge.
Shubham kurutvam kalyanam arogyam dhanasampadah. Shubham kurutvam kalyanam arogyam dhanasampadah. Shatru buddhi vinashaya deepajyotir namostute. Deepajyotir namostute. Deepajyotir parabrahma. Deepajyotir janardanah. Deepajyotir parabrahma. Deepajyotir janardanah. Deepo.
Thank you all. Now it's my privilege to invite Dr. Gopinath Sir, Organizing Secretary, BMS Econ 2026, to deliver the welcome address and the theme of BMS Econ 2026. Sir, the stage is yours.
Thank you. With the blessings of our founder chairman, respected Dr. A. Shanmugasundaram, respected Dr. A. S. Ganesan, our Honorable Chancellor VMRF, respected Dr. Anuradha Ganeshan, Honorable Vice President VMRF, esteemed Dr. P. K. Sudhir, Honorable Vice Chancellor VMRF, respected Dr. Mani, Dean Avmc, Honorable Mr. J. Suresh Samuel, Member Executive Council VMRF, respected Medical Superintendent, Vice Dean Admin, Vice Dean Academics, and Vice Dean Student Welfare, other administrators, faculty members, distinguished speakers, delegates, dear students, and participants joining us from across the globe. I, Dr. M. Gopinath, Professor and Head of Physiology, on behalf of BMS Econ team, Organizing Secretary, very happy to give a very warm and pleasant morning to one and all of you. The science of today is the technology of tomorrow. This quote beautifully captures the spirit of scientific inquiry that brings us together today. It is my immense privilege and honor to extend a heartfelt welcome to each one of you to the 6th International E-Conference, BMS Econ 2026, organized by the Departments of Anatomy, Physiology, and Biochemistry, Avmc Puducherry campus. This year's theme, which is Beyond Microscope, Transformative and Future Strategies in Basic Medical Sciences, reflects our commitment to advancing the boundaries of foundational sciences and redefining innovations that translate into meaningful clinical applications. BMSecon has now reached a significant milestone, that is its sixth consecutive year. Over the years, it has evolved into a vibrant academic platform that brings together researchers, academicians, students, and all other throughout the world, enabling the exchange of knowledge and promoting collaborative scientific inquiry. We are delighted to witness an overwhelming response this year with more than 650 registrations and 150 research abstracts, reflecting the growing recognition of this conference as a premier scientific forum. We are also privileged to host a distinguished panel of 15 eminent speakers, including 8 national and 7 international experts from countries such as US, Germany, UAE, Malaysia, and West Indies. And their presence truly elevates the academic richness of this conference. We are privileged to have the presence of around 20 esteemed chairpersons whose expertise and guidance will greatly enrich the scientific deliberations of this conference. We take great pride in continuing our initiative to nurture young talent through Young Research Scholar Awards, encouraging innovation and excellence among faculty, postgraduates, PhD scholars, and undergraduate participants. So, on this occasion, I extend my warm welcome to all the respected administrators, dignitaries, and organizing committee and students for joining this academic extravaganza. I propose my special welcome to our TNMC observer, Dr. T. Prasad, who is a professor of community medicine. I also extend my appreciation to all the faculty members and technical team for their dedicated efforts behind the scenes. As we come together on this virtual platform today, I am confident that BMCCon 2026 will serve as a fruitful forum for knowledge sharing, inspiration, and future collaborations also. With this, I conclude my welcome address, extending my heartfelt and reverential greetings to all our esteemed dignitaries, speakers, and participants present one and all here. Thank you.
Thank you, sir, for your warm and gracious welcome. Every successful conference has a story of vision, dedication, and growth. To take us through the inspiring journey and milestones of BMS Econ, I invite Dr. Rajan sir, Vice Dean, Administration and Events, Avmc.
Thank you, ma'am. I'll just share my slide. Do you able to see the slide, ma'am?
Yes, sir.
Okay. Very good morning to each and everyone who are gathered today virtually for this BMS Econ 2026. I thank the organizing team for giving a wonderful opportunity to talk about the journey and the milestone of BMS Econ. I will be definitely part of this conference because I am the one instrumental to initiate this BMS Econ. Before I start the journey and milestone, I take this opportunity to wish our Honorable Chancellor Dr. A.S. Ganesan, Chancellor PMR of DU Salem, on this day of his birthday, on behalf of all the administrators, faculties, and staff and students. Happy birthday to you, sir. In 2003, Dr. K. Srinivasan sir started one state-level conference at Avmc, where 452 participants were there. And in this conference, by seeing the overwhelming response, our Chancellor sir that time itself made accommodations free and has given you a very good complimentary bag of a big suitcase that at that time I was joined here as assistant professor. And in 2007, Dr. Srinivas again conducted another state-level conference where around 500 to 600 participants were there. And in this conference, our chancellors are made the even conference fees itself free. So he has refunded all the conference fees at that time by seeing the voluntary response of the participant. So in 2018, after long gap, so once we have taken over the department of the anatomy, so we were the youngster that started thinking. So those days in the beginning of the institute started when K.R. Srinivasan sir can able to organize a wonderful conference. Why not? And if at all if you are organized, why not to plan for a international conference? That is our motive. So we have planned an international conference, a physical mode, which was successfully conducted in 2018 with the 12 speakers, and 8 were international speakers, and the 612 participants were there. After that, so we were thinking that so there is a response for the conference which is organized by the Department of Anatomy and AVMC. So why not we plan for a BM basic medical science altogether? Why not to plan this, and why not to continue this as a continuous event? That's what we decided. So we were planned to have a physical conference that time. So with all the 3 departments. So but unfortunately, the COVID-19, so which were made as to conduct a online conference. Immediately, we just planned a online conference. Then after that, we are made it as a continuous event. So we have organized another conference in 2020. Then we organized 2022, Dr. Lakshmi Sati organized the conference with 16 speakers and 807 participants. Then 2023, Dr. Manju. On rotation, we were the organizing secretary has been keep changing. Dr. Manju organized the event in 2023 with the 15 speakers and 831 participants. And followed by Dr. Ilangadir organized in 2024 of 12 speakers and 536 participants. And now in 2026, we are here, and Dr. Gobinath is organizing this event with the 15 speakers and 615 participants and 155 abstracts submitted for the presentations. So this is about our journey and milestones. These are the glimpses of the conference which was started from 2003 to 2026. So these are all the physical conference glimpses where our Honorable Chancellor is lighting the lamp and honoring the Chief Minister and honoring the Minister and honoring the delegates. And Dr. K.R. Srinivasan honoring Dr. A.K. Krishnamurthy, so one of the renowned faculty of the anatomy. And Dr. K.R. Srinivasan with his madam. And this is an international conference. These are the international speakers who came from Singapore, Malaysia. And these are all the speakers. And this is about the participants who participated in the various conferences. And this is the session who chaired them. And this is a certificate distributed to the participants as well as to the resource persons. And this is about the photo taken by the delegates. This is a sit-chat movement of our organizing team. This is the cultural events which we have conducted. And after that, we move on to the virtual mode. So this is a virtual mode conference, I can say. These are faculties, and these are the participants who have taken a virtual photograph, group photo. And the souvenir released, even though the COVID, we released the souvenirs. And this is about the group photo. Thank you for this opportunity. Thank you all.
Thank you, sir, for that insightful overview of BMS Econ's remarkable journey. It's now time for a significant moment of this ceremony. I respectfully invite respected Mr. J. Suresh Samuel, sir, Member, Executive Council, Vinayaka Missions Research Foundation, to declare the conference open and to address the gathering.
Good morning, all of you. Hope I'm audible.
Yes, sir.
So it's my privilege and pleasure to declare open the 6th International BMC Conference 2026. Incredible progression that we have made year on year. What I heard from Dr. Rajan over the last 6 years in conducting this conference and bringing in important topics for discussion, which is more relevant in the current days. At this important juncture, where technology gets integrated more with medical education and research, the foundational sciences of anatomy, physiology, and biochemistry are assuming a renewed and transformative role in shaping personalized, and precision medicine. Recent advancement in anatomical sciences, including digital imaging, three-dimensional modeling, patient-centric and specific planning, and redefining how structural knowledge supports the individual care are becoming an important topic. While one side is there, like what we heard from Dr. Parthi on the works that has been happening in Vinayaga Mission, AVMC, with regard to the digital twin, digital pathology. The another concern is more in terms of how Dr. Deepthi brought up in terms of what should be the necessary validation, how we really need to ensure that the hallucinations of the AI doesn't really shadow in terms of what the real protocol and treatment which has come through the experience of so many years in the field of medical, how that integrates. So all these things are very important topic for discussion. Modern physiology is increasingly guided by predictive analysis, real-time monitoring, and data-driven understanding of human function, helping clinicians tailor interventions with greater accuracy. And when it comes to biochemistry, it continues to drive this evolution through genomics, biomarker discovery, and pharmacogenomics, enabling therapies that are more precise, effective, and responsive to individual variation. I'm sure this BMSC Con 2026 reflects the contemporary vision by bringing together the scholars, clinicians, educators, and researchers committed advancing the frontiers of foundational medical science. Such platform like BMSC Con provides an avenue for academic exchange, but also an incubation for innovation, interdisciplinary collaboration, and meaningful scientific progression in healthcare, particularly in this current trends of artificial intelligence and technology integrated or driven medicine. I wish this conference a great success, and I congratulate this team for holding this conference at a great visibility globally for the consecutive sixth time. Thank you. All the very best.
Thank you, sir, for your inspiring words and for officially declaring BMS Econ 2026 open. I now invite Dr. P.K. Sudhir, sir, our Honorable Vice Chancellor, Vinayaka Missions Research Foundation, for the release of the e-souvenir of the conference.
With utmost happiness, I release the souvenir of Basic Medical Science e-Conference 2026.
Thank you, sir, for unveiling the conference souvenir, and I also humbly request you to address the gathering, sir.
Respected Dean Avmc and President of this today's program, Professor R. Mani, Honorable University Executive Council Member, Mr. J. Suresh Samuel, our Vice Dean, Professor Rajan, our Organizing Committee Chairperson, Dr. Manju, Organizing Committee Secretary, Professor Gopinath, distinguished guest, eminent speakers, respected faculty members, dear researchers, students, participants, ladies and gentlemen, a very, very good morning to you all. It gives me an immense pleasure to welcome you all to this 6th International e-Conference by Basic Medical Science e-Conference 2026, organized by the Department of Anatomy, Physiology and Biochemistry of AVMC, a constituent college of Vinayaga Mission Research Foundation deemed university at Puducherry campus. At the outset, I congratulate the organizing team for conceptualizing the theme that is both timely and visionary, beyond microscope, transformative and future strategies in basic medical sciences. This theme beautifully captures the evolving landscape of medical education and research, where we move beyond conventional observation to innovation, integration, and impactful discovery. Vinayaga Mission Research Foundation deemed university always encourage and support both technically and financially to organize conferences and workshops for the benefit of the stakeholders. Basic medical science form the very foundation of healthcare. Today, these disciplines are no longer confined to the textbooks or laboratories. They are expanding into domains of such as molecular medicine, artificial intelligence, translational research, and precision medicine. The microscope today is not just an instrument. It symbolizes curiosity, exploration, and the quest for deeper understanding. In this context, this conference serves as an important academic platform to exchange ideas, present research, and foster collaborations across disciplines and borders. It is through such interactions that we can bridge the gap between basic science and clinical application, ultimately improving the patient care and public health outcomes. I'm particularly pleased to note that this is an international e-conference enabling participation from experts and scholars across the globe. This reflects our institution's commitment to global engagement, academic excellence, and knowledge sharing. I would like to say to the young researchers and students participating in this conference, this is your opportunity to think beyond boundaries and contribute meaningfully to the future of medical science. Innovation often begins with a simple question.
Hello.
Never hesitate to ask, explore, and experiment. I also take this opportunity to appreciate the relentless efforts of the faculty and the organizing committee, whose dedication has made this event possible. Your commitment to academic excellence continues to elevate the standards of our institution. As we inaugurate this conference, I am confident that the deliberations over the next 3 days will be intellectually stimulating and professionally enriching for all the participants. I wish you all a fruitful deliberations and a rewarding conference experience. Thank you.
Thank you, sir, for your motivating and insightful address. I now request Dr. R. Mani, sir, a respected Dean, AVMC, to deliver the presidential address. Over to you, sir.
Thank you. Respected Executive Council Member, VMRF, Mr. Suresh Samuel, sir. Respected Vice Chancellor, VMRF. Respected national and international dignitaries, scholars, and students. It is a proud moment and a privilege also to deliver the initial address at the 6th International Conference, e-conference of BMCCon 2026, organized by the Departments of Anatomy, Physiology, and Biochemistry. This conference themed Beyond Microscope, Transformative and Future Strategies in Basic Medical Sciences only reflects our readiness to embrace innovation in medical education, research, and clinical practice. I am delighted to note that this year's conference has attracted around 650 registrations, representing a vibrant mix of faculty members, researchers, postgraduate students, PhD scholars, and also undergraduate students. This enthusiastic participation stands as a testimony to the growing relevance of basic medical sciences in shaping the future of healthcare. The current edition of BMCCon continues this tradition with a rich scientific program that includes 16 eminent international and national speakers, cutting-edge topics such as artificial intelligence in medicine, translational research, and innovative teaching methodologies. I'm happy to see the involvement of the delegates from the number of abstracts we have received for the scientific oral and poster presentations, which is around 150 abstracts. Another highlight is Young Research Scholar Award for all categories with a total cash prize of rupees 25,000. At this moment, I thank the management for giving an opportunity in organizing the BMCCon for the 6th time. I would like to appreciate the dedicated efforts of the organizing committee, the Faculty of Anatomy, Physiology, and Biochemistry, and all the stakeholders who have worked tirelessly to bring this conference to a reality. I am confident that BMCCon 2026 will inspire meaningful discussions, stimulate innovative ideas, and strengthen the professional network. I wish the conference great success and all participants a truly enriching academic experience. Thank you, sir.
Thank you, sir, for your inspiring leadership and guidance. I now invite Dr. Damodran, sir, our respected medical superintendent, to address the gathering. Over to you, sir.
Very good morning to all my dear dignitaries, faculties, and students, the participants. First, I should congratulate the team having organized the 6th International E-Conference for the Basic Department of Anatomy, Physiology, and Biochemistry with the theme of Beyond Microscope, Transformative and Future Strategic in Basic Medical Science. The team headed by Professor Dr. Gopinath, head of the Department of Physiology, and his team effectively gathered 615, I think, today's census of a registered person might enhance also the next 2 days. So this is the platform to have an exchange of knowledge at the in international level, at the students level, with all the participants of various universities, various institutions. So it's a better platform in the future also. I hope that the students of the various categories of the basic medicine will have an entry, they can enrich our knowledge through this platform. I hope this platform will continue for the future medical industry, by which the clinical strategy and clinical knowledge, now that AI is also engulfing all the departments going to be. So unless we have a basic knowledge adequately, we cannot survive in the medical industry. I hope this type of e-conference will give a better future for all medical fraternity. I hope it will be a best successful e-conference. We keep continuing this platform. And once again, congratulate the entire team headed by Dr. Gopinath having organized. I hope it will be very useful in future also. Thank you so much.
Thank you, sir, for your valuable insights and support. Gratitude is the fairest blossom of all. I now invite Dr. Manju ma'am, organizing chairperson, to deliver the vote of thanks. Over to you, ma'am.
Thank you, Dr. Kalaivani. A very good morning to one and all. Respected Honorable Chancellor, Dr. A.S. Ganeshan, sir, Vice President, VMRF, Dr. Anuradha Ganeshan, ma'am, Vice Chancellor, Dr. M. Sudhir, sir, Mr. Suresh Samuel, Member of Executive Council, respected Dean, respected administrators, and all distinguished dignitaries, esteemed speakers, dear faculty members, beloved students, and my dear delegates. It's my proud privilege as the organizing chairperson of BMS Econ 2026 to propose the vote of thanks for this inaugural session. First and foremost, I would like to thank God Almighty for the blessing showered upon us, which helped us give a shape to our conference and make it happen today. I want to express my heartfelt gratitude to our founder chancellor, late Dr. A. Shanmugasundaram sir and Annapurni Shanmugasundaram madam, whose blessings paved the way for conducting this mega conference. At the outset, I express our profound gratitude to our Honorable Chancellor, Dr. A. Ganeshan sir, and Dr. Anuradha Ganeshan ma'am, the Vice President of PMRF in absentia, for their continued guidance and unwavering support. Thank you, sir and ma'am. Today being sir's birthday, we wish you a very happy birthday, sir. Although sir and madam could not be with us this morning, their best wishes, consistent support, and blessings, they have always played an important role in shaping this conference and making it a success. My sincere thanks goes to our beloved Vice Chancellor, Dr. P.K. Sudhir sir, who has always showed interest and supported us in conducting our conference. Thank you for joining us and addressing the delegates, sir. I also take this opportunity on behalf of the organizing team to extend my sincere thanks to our respected member, Executive Council, Mr. J. Suresh Samuel, sir. He is the driving force behind all the academic events in our institute. Thank you, sir, for your constant encouragement and support in organizing this conference. Your support has given us a framework upon which we have planned and executed this program. We thank you for joining us today, sir. Now, my heartfelt thanks goes to our respected Dean, Dr. R. Mani, sir, for the presidential address delivered. His constant encouragement, unconditional and overwhelming support, and timely advice has made this event possible. Thank you very much, sir. Next, our sincere gratitude to all the administrators, Dean Biomedical Research, Dr. Rakesh Sehgal, Medical Superintendent, Dr. Damodaran, sir, for the constant support and inspiration. I would like to extend my sincere thanks to Vice Dean Admin, Dr. T. Rajan, sir, who is one of the organizing chairpersons too, along with me, for making us to recollect the path we walked through to reach where we are today through the history and growth of our conference. Thank you, sir. I extend my gratitude to Vice Dean Academics and Digitalization, Dr. Lata, ma'am, for all the support offered. Thank you, ma'am. On behalf of the team, I extend my thanks to the Vice Dean Student Affairs, Dr. Lakshmi Jatia, ma'am, Deputy Medical Superintendent, Dr. Prem Kumar, Dr. Shanmugavalli, Dr. Arunachalam, sir, Deputy Director at GOS, for the unwavering support in organizing this conference. I also would like to express my heartfelt gratitude to our very esteemed speakers, chairpersons, and judges who have consented to support us in this venture. Thank you all. I also would like to thank the heads and staffs of our other colleges under VMRF and heads of the departments of AVMC for their valuable support. Thank you all. I sincerely thank our TNMC observer for our conference, Dr. T. Prasad, Professor, Department of Community Medicine, for being with us today and supporting us. Thank you, sir. I thank all our enthusiastic delegates who have dedicated their valuable time by showing interest upon registering for our conference, participating live today, sharing their study abstracts, and making our conference a benchmark in the history. Thank you all, dear delegates. I take this opportunity to extend my sincere appreciation to the organizing secretary for enlightening the theme of the conference, and a very, very special gratitude for the entire organizing committee for their meticulous planning and tireless efforts behind the scenes, which made this conference happen today. Now, finally, I would like to express my gratitude to the finance department, technical team, support staff from publication division, EDP, audiovisual section, informatics section, housekeeping, electricity division, the virtual platform, event management team, meetings and more for ensuring the smooth conduct of this virtual inaugural session. With this, I once again thank each one of you for your presence and contributions. We look forward to your active participation in the scientific sessions ahead. Thank you, one and all.
Thank you, ma'am, for that heartfelt expression of gratitude. Finally, let us rise for the national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा चञ्चल जलधितरङ्गा तव शुभ नामे जागे तव शुभ आशिष मागे गाहे तव जय गाथा जनगण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you all for being a part of this inaugural ceremony. We wish you a fruitful and enriching conference ahead. With this, we officially commence BMS Econ 2026. Let us engage, explore, and innovate together as we move beyond the microscope into the future of medical sciences. Thank you all.
Yes. Good morning, everybody. We'll be starting our scientific session, the first session now. Give me a few seconds, please. M&M team, can you please check whether chairperson, madam, has entered, Dr. Vidya? Good morning, Jayprakash, sir.
Yes, ma'am, I'm here.
Okay, ma'am. Can you please switch on your videos too? Okay.
Good morning, Dr. Manju.
Yes. Good morning, sir. Good morning, ma'am. So we'll start our first session of, the second session, in fact. First one session got over before our inaugural session. So it's it's my pleasure and it's my pride to start this. I'll be moderating this session, sir. So it's my pleasure to first introduce our chairperson for this session. Before I introduce the chairperson, I would like to personally wholeheartedly thank both of you, sir, Dr. Jayprakash, sir, and Dr. Vidya madam for accepting this to be a speaker and as a chairperson for this conference. Thank you very much, sir, and thank you very much, ma'am. So it's my privilege to introduce the chairperson for this session. It's Dr. Desai Vidya Sripad, ma'am. She's a professor and head Department of Biochemistry from All India Institute of Medical Sciences, Mangalagiri, Andhra Pradesh. She has finished her MD from Government Siddhartha Medical College, Vijayawada. And she has, looking into her academic and professional excellence, she's an assessor of NABL, as well as for CII, Confederation of Indian Industry, the Educational Excellence Program and Baldrige Excellence Framework too. She's also a state representative for AMBI, and she's a principal member of Bureau of Indian Standards, BIS, and Medical Laboratory Quality Management Panel too. And her scholarly contributions are many. She has made many presentations, but the main highlights being the platform presentation Madam has made in Australia in 2024 in the Asia Pacific Federation of Clinical Biochemistry Conference, the poster presentation in Chicago in 2025 in ADLM, and she has multiple publications, around 22 publications in various index national and international journals. Then the next slide, please. Then her topics of research interest include the oxidative stress markers, PMS, PCOS, metabolic syndrome, yoga, music, dance as a holistic approach to treat Alzheimer's disease. And among the achievements, she's Power Woman Award for 2021. Ma'am has grabbed it as a part of the world record in honoring 111 women in 111 minutes by the Lead India Foundation. She's a woman. She was awarded the Women Leader Award for 2019 by Transformance Business Media. And she has been a keynote speaker of multiple conferences, the main being International Conference on Multidisciplinary Research and Innovation, Dubai in 2023, on the topic Advances in Medical Field, then Mara University Conference, Future of Medical Genetics. And ma'am has also been one of our speakers in the previous BMS Econs. Welcome, ma'am. And you can take over from here, ma'am. Over to you, ma'am. Vidya, ma'am. Vidya, ma'am.
Yes. Good morning.
Yes. Yeah, the slides, please. The slides, please. Yes. Ma'am, you may go ahead, ma'am. Ma'am, you're muted. Vidya, ma'am, you are muted, ma'am.
Yes, 1 minute. Am I audible?
Now you're audible, ma'am. You may go ahead, ma'am. You can introduce.
I take this opportunity to introduce Dr. Jaiprakash Mohan Raj. A very good morning to all of you. And PMSE Con 2026, a huge good luck for all of you to start this academic event. Dr. Jaiprakash, he is MD PhD and PG Diploma Medical Education, Associate Dean for Student Affairs from International Medical University, Kuala Lumpur, Malaysia. He has done his MBBS, MD in Biochemistry and PhD from University of Dundee, UK. He is a scientist and an academic leader with 20 years of experience. His expertise are in molecular medicine, cardiovascular biology, and health profession education. And his scholarly highlights are he has Q1 and Q2 publication and H index of 9 in Scopus, and he was an international presenter in APFCB 2024 Sydney. And over to you, sir, for your...
Please.
Yes, I'm sorry. His research focuses are, today he's going to talk on striatin to all of us. Other research focuses are molecular signaling mechanism in cardiometabolic and vascular diseases, mainly on emphasis on cardiovascular and cancer interface. He has recent scholarship work on striatin as a key signaling hub, which is linking vascular homeostasis and oncogenic progression. He has a funded research in adipokines, oxidative stress, metabolic biomarkers, and cardiometabolic risk profiling in multi-ethnic population. The other academic innovations, leadership, and contributions are he has played a major role in curriculum development and program accreditation. He is a leadership in assessment systems and academic governance. He has pioneered AI-driven feedback and structured remediation frameworks in medical education. His scholarship bridges bench science, clinical relevance, and academic leadership, and which contributes to interdisciplinary biomedical research. So, to Sir, on his topic, striatin as a signaling hub, bridging cardiovascular biology and cancer, we are very eager to hear from you, sir. Over to you.
Thank you, Dr. Vidya. Well, I'm very happy to see you again. I think the last time we both met was in Sydney.
Yes.
We had quite an interesting conversation over there. I do remember.
Yes.
Well, at the outset, let me begin by thanking Dr. Manju for helping me associate with this particular organization and conference. Thank you, Dr. Manju, for the opportunity. And of course, to BMSC Con for giving me this platform to talk about our research. Okay, to begin with, I just want to also introduce that this is not just my research; it's my team's research, and along with me, I have Dr. Shahdi. She's also a colleague who brings a wealth of experience and expertise to this particular work that we have done. So let me share my screen to begin the presentation. Dr. Manju, I would like to know how much time do we have in terms
so they'll
of...
switch on the, now the timer will be switched on, sir, so 35 minutes. You'll have 5 more minutes extra after this.
Okay, so I have 30 minutes to complete the presentation, right?
Yes, sir.
Okay, thank you so very much. Just confirming with my presentation, is it visible?
Yes, sir, you can make it full screen, sir.
Yeah.
You can make it into the presentation modes.
Done. Is it now visible?
Yes, yes.
Okay, so let me quickly. Is the presentation mode off or on?
Presentation mode is gone, sir. Please.
or you want me to bring back the presentation mode?
Yeah.
Okay, how about now?
Yes, sir. Now it's in the presentation.
Okay, fantastic. Thank you so very much.
Thank you.
All right. So good morning to all gathered here. I'm so happy to have this platform to present our work, a paper basically on striatin, a multifunctional protein in cardiovascular and cancer pathology, which is the direct foundation for this presentation. Now, we often link heart disease and cancer as a completely separate conditions, but today we'll see how just one protein, the core striatin protein, can act as a critical junction in the cell and how disrupting its blueprint can have a devastating consequence. I'll be taking you through the fundamental biology and the molecular architecture of this particular protein, striatin, and also a bit on StriPac, while my colleague, Dr. Shadi, will detail on the specific clinical connections. Before we begin, a short insight on the team. I hope I'm still on the slide. Am I visible on the slide? Just thought I should.
Yes.
My slide is still visible? Okay, because
Yes, sir.
just give me a minute because I lost the slide.
It's visible.
Okay, give me a minute. All right. Yeah, there you go. Sorry about that. I'm having a bit of a technical challenge. Just give me 1 minute. All right, sorry about that. Okay, so here you go. We can continue from here on. Okay, so this is our team who were part of this particular study. So Dr. Shadi is a senior lecturer in physiology and also a chair for school research. She works along with me in IMU University. We have broken down this presentation into 2 sections where I'll be talking on the molecular aspect of this particular protein, while Dr. Shadi brings in the connection between between its effect on these clinical conditions. All right. Moving ahead, a bit about IMU. Now, we both are associated with IMU, and for those who are less familiar about our institution, it's one of those premier private institutions in Malaysia that was established way back in 1992, and we are known for our global partnerships. We have about 40 institutions with which we work across the globe as partner universities, and our One Health vision has brought in many collaborations. And I'm proud to represent the research output coming from this institution. All right, now let me begin the first part of this presentation titled Deconstructing StriPac, the Molecular Architecture in Cellular Process. All right. Okay, now what you see over here,
the slide is not changed. So it's still the first slide, sir.
Oh, it's still the first slide. Okay, let me stop sharing and start reshare again.
Please,
Looks like there's kind of a technical issue. Okay.
Yes, sir. Now you start.
Now it's okay.
Yeah,
all
make
right.
it full presentation mode, sir. Yeah, now it's seen, sir. You may please.
Thank you very much for that.
Thank you, sir.
All right. Okay. So as you can see in the diagram on the right, striatin acts like a master architect. Physically, it brings different proteins, key proteins like phosphatases and kinases together. Now, think of it as providing a precise structure that transforms the random molecular collisions that happen within the cell into a highly tuned signaling pathway. And now what this allows is it ensures that the messages travel precisely along the correct tracks. Now, striatin and its organization ensures that the cellular chaos that is usually seen is brought into a certain kind of an organizational structure, preventing critical functions or facilitating critical functions like growth or movement, and directing them on how they should be, how they are formulated or delivered. So it's more like a main player organizing or bringing order to this particular chaos. Now, to illustrate how essential this molecular architecture is, look no further than this particular table. Now, what we see that the core design of this stripac complex has been preserved across millions of years of evolution, right from mammals to fruit flies or even fungi. Right now, nature has found a highly effective scaffolding blueprint, and it has stuck with it for ages. Meaning that the way it controls the signaling and transport, it's truly fundamental on how life works at this cellular level. Now, in this, let's look at the blueprint itself. Okay, the stripac modular chassis has got many key domains. All right, the cavelin binding domain is one of the first and important ones, which acts like a postal code, if you may. Now, what it does is it anchors the protein complex to the membrane. That's one part of this blueprint. Now, without this precise anchoring, where would this protein complex even begin? So that's essential in this architecture. Now, the next one is your coil domain. It acts like an engine, allowing different proteins to click together like a precise Lego block. So it brings in and interlocks them together. Then the calmodulin domain serves as a sensor for calcium, and this kind of interacts with the environmental factors, allowing the cell to respond appropriately. And then we have the WD40 repeats, which provides docking way for other crucial proteins. Now, we know this is backed by extensive research from Cassettes, Bartoli, and Koch. They all provide well-documented evidence that represents this particular model. Now, let's look at, now, with this understanding of this molecular blueprint, how do we actually build that molecular machine? Now, from the blueprint, the way we understand this molecular machine. Now, think of it as a modular assembly line. All right. Now, it starts with the core dimer. Now, as two striaton molecules combine, they form the core dimer. Right. Now, once these two striaton molecules form the dimer, then they come together by an adapter bridge. Now, this adapter bridge attaches crucial proteins like your CCM3 and docks it into the striaton scaffold. Right. Now, remember, even if one pieces are connected poorly, the entire signaling breaks down. So that's how important this assembly bridge is. Now, after that, the chassis anchoring, once that chassis anchoring is secured to the membrane, you will have to bring in powerful enzymes, without which the foundational core is of no use. So that's where the catalytic payloads are brought in terms of the germinal center kinase protein being anchored to it, or phosphatase PP2A being anchored to it, right? Now, once all of these are brought in, finally, the functional accessories like your eNOS and other proteins like your MOP3, they all are plugged in. Now, remember, every step is crucial for the functional complexity. Now, striaton in that context acts like a specialized docking station, right? Now, even though it does not have intrinsic activity on its own, by physically bringing together enzymes like phosphatases and kinases, it forces a very precise and controlled reaction within the cell. Now, it turns the random cellular chaos into a more organized messaging. And it acts like something like a switch that directs the signal. Okay, when should the phosphatase be turned on, or when should the kinase be turned on? Now, this mechanism is very heavily documented by Maroney and et al. And Gardner and et al. And they highlighting its role as they call it as a very critical molecular switch. This particular picture or image kind of gives you an illustrated view of how all these elements are brought together. Now, if you notice, you have the cavelin protein, which is binding to the cavelin domain of striaton, and you can see the dimerization of 2-striaton, facilitating for the adapter bridge, for the key enzymes, PP2A coming together, the GCKs, and then you can also see the enoses. All of these in turn are going to trigger multiple pathways. So this kind of gives you an idea of what that STRIPAC complex looks like. All right, now moving ahead, we are going to talk a little bit of the calcium toggle state. Now, this slide highlights the system's dynamic control. Now, think of calcium like a toggle switch, right? Now, when levels of calcium are low, striatin remains anchored to the membrane. But when the calcium floods into the cell or the cytosol, it binds to the calmodulin domain, which we earlier reflected on. And this causes the complex to detach and retreat into the cytosol. Now, this toggle state, as described by Hong and Tallis, and they demonstrate how this entire system controls, the cell controls this entire system dynamics in real time. So the way in which they change their state is very specific to the effectors that act upon it. In this case, calcium. Now, as you can see, for that particular state to exist, being from anchored to the non-anchored state, you need something like caveolin binding domain. Now, as you can see in this particular slide, striatin requires a precise address, as in the caveolin binding domain. Now, this domain is designed to recognize and attach a specialized lipid raft. Now, the key mechanism for this specific FLQHEWARF motif. This is the amino acid sequence, the motif. The explicit sequence requires it to bind to the caveolin 1 on the cell membrane. Now, if you were to knock off this particular sequence of amino acid, striatin would lose its ability to target the membrane, disrupting the whole scaffolding ability. Right? And this is very well highlighted in the paper by Gordon and Cassettes. Moving ahead, we are going to look at how this entire molecular switchboard, or master switchboard we call, is associated with signaling network. Now this kind of gives you an idea of how the whole central network is brought together and what impact does it have. Now, if you can think of STRIPAC like a cell's master switchboard, right? By physically linking critical pathways like Hippo pathway, it can regulate organ size and tumor suppression. Or by MAP kinase, it regulates cellular proliferation and differentiation. Right? Now, it acts like a central hub. Now, any disruption to this STRIPAC assembly, as documented in the paper by Endo et al. Or Huang, can throw the entire network into disorder, now creating a very disastrous ripple effect across the entire cell. So that's the importance of the STRIPAC complex. I'm going to give you one example of how it is all brought together. Now, here we see how this striatin bridge provides a scaffolding for a rapid non-genomic steroid signaling. Now, most of the time, we notice that certain signals takes a few hours to minutes to send a signal down. But in certain cases, it needs to reach the cell much faster. Now, instead of pathways taking long hours to affect the gene, striatin bridges, in this case, estrogen receptor, alpha estrogen receptor, directly to enos. So it acts like that kind of a scaffolding protein to enable a rapid burst of nitric oxide. Now, similarly, it functions as a hub for aldosterone, where rapid signaling impacts kidney function and cardiovascular health, as shown by the paper by Pogo. Now, moving on to another example related to cellular logistics in terms of, in this case, transport. This slide shifts the focus from the STRIPAC complex or striatin acting more of a scaffold protein to a more cell chemical signaling to physical logistics. Now think of striatin as a cell's delivery manager. Now, in this picture, I'm trying to capture that essence. Now, what striatin does is it coordinates cellular movement. Like we all have heard about this clathrin-dependent endocytosis, right? We call it cell pinching off. Now, as Schultz projected in their paper, or have shown in the paper, in neurons, you need striatin acts like those elements, they're like those through those dynamin 1 proteins help the clathrin coated pits to pinch off, enabling endocytosis. Now, along with that, what it also does is it acts as a molecular tether using the WD propeller. Now, what this does is it connects to the cargos, like as shown over here, autophagosomes, to the dynein molecule. So that kind of a bridge it creates, and it facilitates essential transports along nerve fibers. So that's how it kind of facilitates axonal transport. So all of these is kind of showing you the importance of striatin, its role in cellular physiology, and how it functions at a molecular level. Now, it ties me down to the final few slides where we can see that certain clinical pathologies, like this final slide, it kind of shows you what happens when this architecture fails. Now, for example, over here, if the striatin gene is disrupted, or there is an error in the gene, it can lead to fusion proteins that can cause unregulated growth in lung and thyroid cancers. Now, problems with adapters like CCM3 that we earlier saw can cause brain vascular lesions. And variations in the core protein can also disrupt cellular signals, leading to hypertension, heart failure, or Brugada syndrome. So these are some of the impact of disruption of either the protein or the complex. So in summary, I would like to conclude by saying that we have covered a complete architectural masterpiece in this case, from modular chassis design to the dynamic assembly that is triggered by calcium, and also we've seen how StriPac acts as a master regulator of systemic health. Now, overseeing everything from tumor suppression to cardiovascular function. As we have seen this in understanding this entire architecture, we are now well placed to take this entire understanding into reflecting on how they play a role in understanding its role in cardio oncology, right? So this is where I pause, and I would like to invite now my colleague and friend, Dr. Shadi, to take over this presentation and engage the audience. Dr. Shadi, over to you.
Welcome, Dr. Shadi, ma'am.
Hello, everyone, and thank you. I would like to thank Dr. J and also the organizing committee for giving me the opportunity to discuss about striatin. Just I'm trying to move a couple of windows around and put this on presentation mode so we can go through. Okay. Do you see the slide?
Yes, ma'am. Yes, ma'am. It's in
okay, great. Thank you. So as Dr. J mentioned, he talked about striatin structure and how it works as part of the StriPac complex. I'm going to continue striatin role in various conditions. In particular, what we are interested in is the cardiovascular and cancer, and see if it can act as a bridge in cardio-oncology. The overall striatin story started for me since I joined Dr. Williams' lab at Harvard a while ago, and since then it has been very close to my heart, and I'm interested to learn and research this protein frequently. So Striatin is essential in mediating mineralocorticoid-dependent rapid response to aldosterone. Previous research showed that striatin expression increases in response to aldosterone. So in our lab, we show that if we treat the cell with aldosterone, we can see that there is increase in downstream aldosterone non-genomic pathway. Like, for instance, there is increase in phosphorylated ERK. So knowing that aldosterone can increase striatin, we decided to block the striatin with striatin siRNA. And as you can see here, blocking striatin siRNA reduced phosphorylated ERK level. However, we didn't see the same response when we used a scrambled siRNA. Another non-genomic pathway that is might be mediated by striatin is that we know aldosterone can increase ROS level. So in the cells that are treated with striatin siRNA, we notice that there is reduction in ROS level. So these are among various different information that confirm basically striatin is the mediator of mineralocorticoid receptor pathway. There are other publications and other research group, in particular from Tufts University, that they showed striatin can mediate also estrogen and estrogen receptor pathway. So looking at one of the publications from this research group, what they found is that estrogen and aldosterone, they can compete for binding to striatin. The question started, if striatin bind to mineralocorticoid receptor, also if estrogen receptor alpha bind to striatin, what will happen if we have both aldosterone and estrogen in the environment? So you can see in this immunoplot here, if the cell are treated with aldosterone, we will see striatin significantly bind to mineralocorticoid receptor. But however, if the cell are treated with estrogen, we can see that striatin binds to estrogen receptor alpha. And you can see basically the calculation and chart on C and 2 plot in the C. But if we treat the cell with aldosterone followed by estrogen, we can see that binding of striatin to MR is reduced. Then in another study, what they did, we already know that there is a peptide that can block striatin binding to estrogen receptor alpha. So what will happen if we block the striatin and estrogen receptor alpha interaction, and we treat the cell with both aldosterone and estrogen? As you can see here, when we block the cell with the peptide, still we have aldosterone binding to MR binding to striatin. However, we don't see any estrogen binding to striatin at this point. So this is among a couple of other evidence that I don't have time to go through it. It shows that there is competition between estrogen receptor alpha and mineralocorticoid receptor to bind to striatin. So if we want to put all of that together, we can summarize it here. Basically, whenever we have aldosterone mineralocorticoid receptor, striatin is going to help to mediate the non-genomic pathway through increase in the PERK pathway, cardiac hypertrophy. But if we have estrogen and estrogen receptor mediated pathway through striatin, we can see that there is increase in phosphorylated enos, which is important in vasodilation, and it has vasoprotective effect. On the other hand, if we treat the cell by estrogen followed by aldosterone, we can see that there is increase in ERK level, extracellular signal regulator kinase again. And vice versa. If we first treat the cell with aldosterone, then we treat the cell with estrogen, what we can see here, we can see that there is increase in the enos level again. So we can see the vasoprotective. This is very interesting. There are several studies that they basically looked at how striatin mediate estrogen receptor pathway, aldosterone receptor pathway. Also, there are studies showing that it can bind to GPER, but we do not know what will happen if we have testosterone in the environment. So that's another unexplored area, which can be help us to basically unlock if this striatin playing any role in the cardiovascular diseases that they have huge sex differences. Now that we just briefly talked about the cellular pathway, let's go to the disease part of it. When we wanted to see, as Dr. Joy mentioned earlier, striatin is involved in various diseases. So we wanted to know what is going on out there. We find out striatin is involved in psoriasis. In fact, striatin was among one of those genes that increased in patients with hyperkeratosis. Also, in those that they have depression, using the sample from UK Biobank, it was identified that this particular striatin risk allele carrier, they have higher chance of developing depression if they are exposed to antibiotic. Same goes that there are striatin misvariant associated with age-related, basically hearing loss. In addition, we saw in silico studies that indicate striatin is involved in various cardiovascular conditions like cardiomyopathy, coronary artery disease. But then, if we want, these are in silico analysis, most of them, but the only functional study that is available right now comes from Dr. Williams' lab at Harvard, which one of them is what we published in 2023. And we identified two SNP in striatin gene. They act as a haplotype. We group them as homozygous non-risk allele carrier, homozygous risk allele carrier, and heterozygous risk allele carriers. G0 to G2, basically the number of risk allele carrier increases. What we saw is that in both men and women, there is trend for salt sensitivity of the blood pressure. Just a reminder that salt sensitivity of blood pressure is the difference in blood pressure on high salt and low salt. But then we wanted to know, because of the interaction between striatin and estrogen receptor, is it the same mechanism that causes salt sensitivity in men and women or different mechanism? That's why we looked at their renal plasma flow. We find out in men, there is those risk allele carrier, they have lower renal plasma flow. However, we don't see the same phenomenon in women. But when we looked at aldosterone level, interestingly, we saw that women risk allele carrier, they have a higher aldosterone level. So what we concluded was that in men, salt sensitivity of the blood pressure is because of impaired vascular reactivity in the kidney, which reduces renal blood flow, causes volume expansion, and then result in salt sensitivity of blood pressure. But when it comes to women, there is enhanced aldosterone level that causes volume expansion and result in salt sensitivity of blood pressure. Basically, what we see is that because of this striatin variant, same phenotype, two different mechanisms, which requires different treatment plan. Then we wanted to see, is there any mechanistic at the in vivo level? We did one study in our lab in which we used laser-induced injury in the arteries in striatin deficient mice and also in wild-type mice. What we found that, let's look at this baseline level, wild-type baseline, and striatin baseline. So these are the striatin deficient mice, and what we can see, they have higher thrombus formation compared to wild-type. But then if we add aldosterone to the equation and we treat them with aldosterone, we can see that those striatin deficient mice, in response to aldosterone, basically there is no change in the thrombus formation level, unlike what we have between wild-type and striatin. Basically, striatin deficiency alone is enough to increase inflammation and increase thrombus formation, which also we show that this animal, they do have reduction in the enos level and they have impaired vascular reactivity. In another study that done by different group, they show that in the diabetic rat model, there is increase in the striatin level. This blue bar are the diabetes. And what we can see is that there is increase in striatin model in the right atrium, in the left atrium, and also in the left ventricle. And changes in the striatin level, interestingly, follow the same thing as ANF. which is the marker of cardiac remodeling. So in both of these conditions and what we found in salt sensitivity of the blood pressure, it shows that striating can be a target in cardiovascular disease. At the same time, we ask ourselves, okay, most of cancer patients, they do have enhanced cardiovascular complication. Can we find any information about role of striating in cancer? Because we mainly focus on cardiovascular. So now we wanted to explore cancer. So we did in silico analysis, and we found that yes, in fact, from the available data, we can see that striating is involved in at least 20 different cancers. So we thought, okay, let's take it to different level and we see if there is any published data on that. Interestingly, we found that yes, there are several cancers reported that they have basically pathology related to striating, from thyroid cancer, lung cancer, renal cancer, pancreatic cancer, and so on. Most of them, they are case reports, and the reason for mechanism that is reported in majority of these striating related cancer is that there is striating infusion with anaplastic lymphoma kinase, with striating ALK. So we wanted to see that now there is also a striating fusion with other protein, but ALK basically account for almost 55% of cancer that we see in relation to striating fusion. If we look at just how ALK is involved in cancer, we can see that in the paper that's published in 2023, ALK basically involved in different cancer, from thyroid, gastric cancer, liver cancer, and it can have translocation with different proteins. When it comes to striating, both striating and ALK, both of them are located on chromosome 2. So during the translocation, there is 8 megabaud deletion, and this deletion, it causes striating and ALK to fuse together. So because of this fusion, what will happen? There is enhanced activity of this ALK. Increased in the ALK activity result in activation of various different kinases that they are under basically downstream of this pathway, and all of them, they are involved in cancer, like PI3K, AKT, MAPK, and so on. If we look at what we have here about thyroid cancer, we see that in this particular study, they reported that, and we saw that same thing is happening in different cancer. Whenever we have striating ALK fusion, there is worse prognosis, very poor prognosis, but is very important to identify that particular mutation. Usually it goes through genomic studies, and they have to try with ALK inhibitor to help with the treatment. And in the animal model studies that they generated using striating and ALK, they found that they do have worse tumor progression as well. If we look at another cancer, for instance, let's look at the lung cancer, it still is the same thing. We have the translocation of striating and ALK, which happened between exon 3 of striating and exon 20 of ALK. And in lung cancer, usually we have other translocation as well. In this particular case, they found that striating ALK is the only factor. And unfortunately, this case, because of the worsening of the condition, didn't respond to the treatment and passed away after 6 months. Then in another condition, in the hepatocellular carcinoma, what they found that looking at different cases is not always about striating translocation, but we have cases like HCC that there is increase in the striating level compared to those tissue that they are not tumor tissue. And we want these people, they wanted to see in this study group, do we see changes in the striating level in vitro? That's why they look at various liver cell line, and they found that only in LO2, which is the non-cancer cell line, we have lower striating level. However, other group, other cell line, they have increase in striating level. Interestingly, what they found out is that as we can see in this graph here, there is different level of striatin. So even those that they have reduction in the striatin level, it doesn't affect how they are going to basically proliferate, and it doesn't affect their proliferation. But what is happening is that if we have lower level of striatin, is going to reduce the invasion of the tumor. That's why those that they have higher striatin, they have higher invasion as well. And in another study was reported that in particular in mesothelioma, when we have striatin and ALK, this can act as a biomarker. We see that mainly in the children and in women under 40 years old. So it's not very common in certain cancer that we see this fusion in everyone. So at this point, for sake of time, we just looked at a couple of examples that in cancer, we have lots of striatin, ALK fusion. We can have increase in striatin expression. So if we want to summarize and see what are the potential mechanisms that they work together, we saw that earlier as well, that in the cardiovascular system, it can be interaction of striatin, whether it be mineralocorticoid receptor or with estrogen receptor, which can cause fibrosis, inflammation. It can affect the vasodilation, vasoconstriction. But when it comes to cancer, because of activation of ALK, we can have activation of various cancer-related pathways. Even though we can see that there is potential for overlapping in the pathway, and most of those cancer patients that they receive ALK inhibitor, they develop cardiovascular condition, complication. However, there is not any study based as our knowledge out there to specifically look at striatin role as a cardiovascular complication during the treatment, which is another unexplored area. So to summarize everything, what we saw is that in the cardiovascular disease, as of now, we know striatin can act as a mediator of estrogen receptor, mineralocorticoid receptor, which can alter inflammation, which can alter vascular reactivity, and therefore can play a role in cardiovascular disease. But when it comes to cancer, the main, at least 55% of cases, they have striatin ALK fusion, which is going to enhance activity of different cancer-related pathway, which at the end can change tumor progression and tumor invasion. Therefore, what we can conclude is that striatin, through alteration of all those signaling pathway, in particular steroid signaling, can lead to both cancer disease and cardiovascular disease. However, as I mentioned, there are several unexplored areas which, if anyone interested, we can always discuss and see if there is any potential opportunity for collaboration. And I think that's all. Thank you so much. And if any question, we both will be happy to answer the question.
Yes. Thank you to both the speakers, Dr. Jayaprakash and Dr. Shadi. It was a very insightful presentation. Striatin as a multifunctional scaffold bridging. And as Dr. Jayaprakash said, I love the architectural masterpiece of the cell. All of you agree? It's such a beautiful miracle molecular signaling, yet unexplored. We do speak on space unexplored, but we also can speak on molecular signaling unexplored. So many things. So to conclude, we know that striatin is one potential biomarker along with ALK fusion. It can lead to many other molecular signaling, and it can lead to connected to different signaling pathways and cancer pathways. Over to Dr. Jayaprakash. I have one question. When you said this multiple signaling pathways, usually it is kinases, phosphatases, and calcium signaling. Out of which one do you feel has the most impact?
I wouldn't probably look at it from that perspective as impact generation. I would say depending on how the cell's functionality is. Stripac has to be seen as a machinery which reacts to what the cell needs. In terms of turning on or turning off phosphatase or adding or removing phosphatase, it is basically trying to channelize a certain functionality based on the signal it receives. So I would say that the role of striatin is to bring everything together. And that's where its importance lies in. And this connects so very well with what Dr. Shadi said, that issues with this striatin has been found to kind of disrupt the next molecular mechanism in terms of activation of pathways. That's what we saw, how when it fuses with the ALK, it can cause disruption, and that leads to uncontrolled cellular proliferation, the oncogene being activated. So I would rather look at it from the point of rather than looking at it which is important or not, I would say how it brings the whole machinery together to direct. It's more like the director who says, okay, this is what is required, this is what you need to do. That's how I would like to put it across.
So you say is either a driver or a facilitator?
Yes.
Yes. And what about salt-sensitive hypertension? Any work on this with striatin?
I think that's something that Dr. Shadi has done a lot. Dr. Shadi?
Dr. Shadi, please. Yes.
So, well, when it comes to salt sensitivity of the blood pressure, what I know is that my mentor is the one that has been working on that, Gordon Williams. So what they do for this particular cohort, as I said, this is very specific functional study because the subject receive a specific diet with this measured salt level. So for 1 week, they receive low salt. For another week, they receive high salt. And then during this time period, they are in the hospital. So constantly their blood pressure has been measured. They go through the angiotensin infusion. And that's why we were able to measure the renal plasma flow. And then the sample is connected, the urine sample, blood sample, PBMCs, and they go through the GWAS. So for salt sensitivity of the blood pressure, the focus has been on striatin. There are multiple genes. If you are interested about salt sensitivity, multiple protein, we know that LSD1 is involved in that. ERAP1 is involved in that. Cloto also, there are reports that Cloto is involved in salt sensitivity of blood pressure. And based on the gene, the mechanism is going to be different. For striatin, our first tag SNP that we identified was that we couldn't come up with a mechanism, but we saw that there are differences between the cohort has Caucasian and African American. We saw that there are differences in how they respond. And interestingly, the initial variant tag SNP was higher in the African American. But we wanted to, in particular in the paper that I was in charge, I was interested to see, can we relate this to men and women because of what we know about striatin? And that was, we saw that there is age effect as well, but interestingly, what we saw that there are mechanism related specific in men and women. But whether striatin has been, salt sensitivity has been explored, let's say in relation to kinases, I don't know about that. I know there was one unpublished study on PP2A, but not as part of Stripac, just PP2A standalone.
Yes, thank you, thank you. And one last question to both of you. If striatin picks up over the next decade, what would be its impact on diagnosis, treatment, and prevention?
Well, actually, there's a lot actually been done, but in terms of its diagnostic implications, we can foresee striatin as something that helps us understand not only the progression, but also the outcome of management of in the patient. So I would say striatin is moving towards a more personalized care, more personalized management. So striatin kind of with understanding striatin more, not only are we understanding the pathogenesis, but also can help in managing the patient. That would be my view. Dr. Shadi?
Yeah. So I agree with what Dr. Dry mentioned, but that would be a miracle protein if we can answer.
Exactly.
But something that I think is going to be helpful because most of these studies, in particular those that we did on salt sensitivity of the blood pressure, they come from a SNP. Some of them are, and even those that I mentioned about depression, about psoriasis, most of them are non-functional SNP. So the next logical step would to identify it, or basically to do the in vivo and in vitro study and see what will be the effect of that. And what personally I like to see, and is my area of interest, to see if we can help with striatin related research to answer some of the question when it comes to the differences between men and women in terms of the disease. In particular, if striatin interact with estrogen receptor, we can see that there are vasoprotective effects, but when it interact with aldosterone, it goes to the other way and enhances inflammation.
So any questions to the audience, Dr. Manju, or we can... Thank you so
Can you
much.
see any questions in the chat box, ma'am? Do you
No,
have?
I don't see any questions in the chat box. Yes.
Okay. Oh.
So it was so beautiful morning to have both Dr. Jayaprakash and Dr. Shadi. And congratulations to Dr. Manju. I enjoy this every year. Thank you for the opportunity. Very happy to meet virtually Jayaprakash. We met in Sydney. And Dr. Shadi, we'd love to meet you sometime in India.
Thank you for the invitation, and thank you, Dr. Jaya, for including me in this.
Thank you so much, Dr. Devi.
Actually, yeah,
Organize-
before we end it, I really would like to thank, because the main question which I wanted to ask was already asked by Vidya madam, that was regarding what is the future of this, we would like, and I would like to say that we all would have been really enlightened about the structural basis, the mechanisms, the molecular, the genetic, all the mechanisms which lead to either any diseases like cardiovascular or anything, or various types of cancers, the case reports. So now I think all the people, the delegates who are attending this thing, they are actually now, they would take this molecule up for at least in any way continuing the research with this striatin molecule going ahead. And I congratulate and I thank also both of you, Sir Jayaprakash sir and Dr. Shadi ma'am, for joining this and sharing your research experiences, your findings with in this conference, ma'am. And I really would wish that your studies go ahead too far, so that you provide us with like what Vidya madam told as a diagnostic or a prognostic marker for specific cancers. Thank you so much. And I use this, though it's on a on a virtual mode, I would like to extend my gratitude to Jayaprakash sir first with a certificate of appreciation and a memento, sir. Please. Thank you so much, sir.
Thank you so much, Dr. Manju. Thank you very much.
Next time. I would like to even thank Dr. Shadi Gulami ma'am. Ma'am, you are so kind enough to join Jayaprakash sir and spare this valuable time with us and sharing your very, very valuable research experiences with us. Thank you, ma'am. This is a small token of gratitude, online virtual mode, ma'am.
Thank you so much for having us and giving us the opportunity to participate.
Thank you.
Thank you, and good luck with the rest of the event. Thank you.
Okay, and next I would like to thank Dr. Vidya madam also with her online certificate and a memento, ma'am. Ma'am, you were always there to support us, either as a resource person or for sharing. Thank you so much, ma'am. And wishing you all the best, ma'am. Thanks, ma'am. Madam is organizing secretary. Thank you. Thank you all. Yes, sir.
Yeah. I just want to say a last word. I think the whole thing came about with Dr. Ravi from IMU reaching out
yes,
to
sir.
you.
I forgot to thank Ravi sir is my mentor for FAMA. Yes, Dr. Ravi Shankar sir is my FAMA mentor, and sir was the one who introduced me to, the Jayaprakash sir, and hence all these things happened.
Exactly.
I personally thank sir, but please convey my regards and gratitude to sir.
And also, I would want to open up this opportunity that we here have been working with striatin. Anybody who need to collaborate with us, brainstorm with us on any projects, or even use us as consultants, we are more than happy. In fact, Dr. Shadi has done tremendous work. So we will be more than happy to work with you in the future. Thank you so much for the opportunity, guys.
Thank you, sir, and all the delegates will utilize this.
Thank you.
Thank you, Shadi ma'am.
Look forward to meet all of you at AMBICon 2026 at AIMS Mangalgiri.
Definitely, ma'am. We'll be there.
Thank you.
Thank you.
Thank you. See you. Jai Hind. Thank you.
Thank you.
Thank you.
I thank Dr. Manju ma'am for the wonderful scientific session. Let's break for the refreshments.
Thank you.
The future will be owned by those who are powered by knowledge and wisdom that transcends time. There is one place where such wisdom is imparted, building individuals to build the world. Welcome to
Vinayaka Mission's Research Foundation, deemed university.
The university, with 27 constituent colleges, delivers cutting-edge knowledge across various disciplines. Medicine, engineering, architecture, management, health sciences, dentistry, measuring up to 135 courses of study, all covered across sprawling and student-friendly campuses. At Vinayaka Missions University, students of various disciplines hone their knowledge right up to doctoral levels. At any given point, there are 15,000 students studying in the campuses. With the vision of creating medical professionals who can meet the needs of the Indian and international communities, Vinayaka Missions University has made a significant contribution to the field of medicine. Four colleges and hospitals in Salem, Karaikal and Puducherry not only groom students into medical professionals, but also ensure easy and affordable medical help to the local population. Beyond medicine, the university is a multicultural hub that has students gaining expertise in a wide range of fields. Our engineering courses are designed to give students comprehensive knowledge in all contemporary fields of study. This is strengthened by state-of-the-art infrastructure. Engineering and technology apart, Vinayaka Missions University is known for its courses in dentistry, pharmacy, homeopathy, nursing, allied health sciences, physiotherapy, architecture, arts and science, and management. Our libraries are well appointed and are a digital and physical resource for both students and faculty. Linked to the central online catalogue, students have access to over 145,000 books, 1,000 journals, and over 5,000 audiovisual aids. The hostel facilities at the campuses spell absolute comfort and provide the right ambience for self-study for the students. Our students are thoroughly encouraged in sports and cultural activities. Besides successfully participating in inter-university sports contests, our students have also excelled in cultural meets and festivals.
Bringing minds together in search of endless wisdom. VMU. Building individuals to build a world, building hopes and dreams on heart at VMU. Friends to stay, VMU. Enjoyment to learn and play, showing the way ahead each day at VMU.
Vinayaka Missions Research Foundation, Salem.
Welcome back. Let's move on to the next session of the day. I request Mrs. Soumya Ma'am, Assistant Professor, Department of Anatomy, to take over the session.
Thank you, ma'am. We'll start the session. I, Ms. Soumya Ganesan, Assistant Professor from the Department of Anatomy, Arboretum Medical College, Puducherry, moderator for this session. On behalf of BMS Econ 6th International Conference, I'll welcome you all for the wonderful event. Now we moving on to the scientific talk number 3. So for this session, we're having two eminent chairperson we're having. So first, before I'm just welcoming our chairperson, I would like to thank our resource person, Dr. Kumar Sadish Ravi sir, for accepting the invitation and be a part of our conference. Thank you, sir. Now, I would like to welcome our chairperson, Dr. T. Sivahami ma'am. Madam has completed her undergraduate from Coimbatore Medical College and postgraduate from the Tanjavur Medical College. Madam is a professor and head of the anatomy department, Tanjavur Medical College. Madam has 36 years of experience in government and 33 years of experience in both UG and PG. Madam has published a paper in various national and international journals. Welcome you, ma'am. Next, I would like to invite our second chairperson, Dr. Sulochana Shaktivel, madam. Madam is professor from Department of Anatomy, Jipmar, Puducherry. Madam has completed her undergraduate from Tirunelveli Medical College and postgraduate from the Tanjavur Medical College. Madam has completed her PhD in Vinayaka Mission University, Salem. And Madam has completed postgraduate diploma in health science in diabetology. Madam has published more than 16 research papers in reputed journals and received 3 Best Paper Award. She also research person for the various academic activity. Welcome you, ma'am.
Thank you, Dr. Soumya.
Thank you. Thank you, Dr. Soumya, for the introduction.
Thank you, ma'am. I request the chairperson to commence the session. Over to you, ma'am.
Yes. Good morning, first of all. Let me thank the organizers for giving me an opportunity to chair this session. And it's my proud and privilege to chair this session and to be part of this conference for many years. And I would like to thank Dr. Rajan uh, for introducing Dr. K.R. Srinivasan as a professor. He was my professor, and where he's a stalwart in organizing many conferences. It's my privilege now to introduce the speaker, Dr. Kumar Satish Ravi, sir. Sir, welcome, sir, for this session.
Thank you.
And it was a blast from Jaipur, where you posted the pictures, and the conference was very good from your side, sir. Thank you for your thing. Sir, I think sir needs no introduction, and I will hand over the introduction of Dr. Ravi sir to my student and my co-chairperson, Dr. Sulochana Shaktivel. Over to you, Dr. Sulochana, to have the privilege to introduce Dr. Satish sir.
Thank you, ma'am. Good morning, ma'am, and good morning, sir. It's my privilege to introduce Dr. Kumar Satish Ravi. Actually, he needs no introduction. Everybody knows him. Sir is at present a professor and head in the Department of Anatomy in the NIMS University, Jaipur. And he is adjunct professor in the Department of Anatomy, Kasturba Medical College. And sir is a editorial member in many journals. And he has also authored many books, especially the Snell's Clinical Neuroanatomy. We welcome you, sir, to the session. Over to you, sir.
Thank you so much, ma'am. Thank you so much for the wonderful
Welcome, sir.
introduction.
Welcome, sir, for the session.
Thank you, ma'am. My screen is visible.
Yes, sir, it is visible.
So good morning, one and all. Respected chairpersons, esteemed faculty members, distinguished delegates, and my colleagues. Apart from that, some of the students who are attending this conference. As I was introduced, I'm Kumar Satish Ravi, and it is both, you can say, the privilege and my pleasure to be here today in the 6th BMSC Con 2026 among all of you. And I would like to sincerely thank Professor Rajan sir and the complete organizing team for giving me this opportunity to share and reflect on a subject I can say that continues to evolve at the very, I can say, the heart of the medical science. And today I will be talking on the evolution of histological visualization from classical foundations to translational innovation. Through this lecture, I would like to say that I invite you
to the journey from the early principles of the optics, I can say, and the primitive microscopes, whatever was there in the previously in the primitive stage, I can say, to this remarkable era where we are talking about the digitalization, or you say the digital pathology, or the era of artificial intelligence. When we talk about the histology, I can say that this histology is no longer restricted or confined to the slides, that is the static slides, which usually we see under the microscope, which you can see on this image. I can say that it has transformed into from static to the dynamic, or you say the technology driven discipline that not only bridges the basic science with the, you say, the clinical departments or the clinical application, you say, where a clinician can interpret the various kinds of the diseases, we say, or when we talk about as an anatomist, when we teach some of the structures, or when we talk about our researchers who explores to search again and again, or you say the research in a molecular level or the pathways, we say. So what I wanted to say over here, that this histological visualization will remain a center to all our understanding. And with this perspective, today I will be talking and will be taking you to this journey, the fascinating journey through, you say, the time from the previous era to the present era of the artificial intelligence or the technology. What I wanted to say over here, that whatever the images which have been incorporated in my presentation, I would like to say that it has been taken from the various sources. I do not claim that the images which have been incorporated in this presentation belongs to me. And some of my contributions you can see over here in this image to the anatomical sciences. Now coming to the topic, what I was talking about the histology. So when we see this microscopic study or the histology, when we talk, right, it gives a chance what we can see by the naked eye, right? So this histology gives us a chance to see beyond that and enter into the world of the microscope, you say, or the microscopic world of the body, we can say. So this helps us to understand how tissues are built, you can say, or how these tissues work and how they change when some of the disease occurs in the body or when the disease develops. So in that way, I can say that the histology connects the anatomy, physiology, that is the function, and then you say the development, that is the embryology, and when it is a diseased condition, it connects to the pathology. So you can say that it is a continuous story, you can say, whether it is anatomy, physiology, embryology, or the pathology. So when we talk about this, the medical students, and when we discuss about this histology, so I can say that it is not about only identifying the slides which we identify under the microscope, but I can say that it is about understanding the very foundation of the normal structure, or you say the function, and then we should understand the, until unless you understand the normal structure, you will not be able to understand the diseased condition. So in nutshell, in one line, I would like to say that it is about the understanding of the very foundation of the structure of which we see normally, and then the diseased conditions. And when this advances, right, then we discuss about the various methods or the imaging technologies. So what I wanted to say over here, that during its development, it has advances in the visualization, and apart from that, the technology has incorporated even in the histological sciences, we say. So apart from that, we can say that it is a bridge between the structure and the function. And by focusing on the microscopic structure of the tissues, what we see, the histology helps us in understanding the connection between the gross anatomy and, as I said, the physiology, and then later on when we come across the various disease condition, we can say the pathological conditions. So what I wanted to say over here, that in this way, it serves an important integrating discipline that shows the structure and functions which are closely related to or influences each other. Now, when we see over here in this slides over here, this histology develops the skill of the pattern recognition, I can say. Now, since the students must identify tissues based on the various characteristic features, what we teach the students, that these are the characteristic features to identify a particular tissue, right? So this is very similar to the clinical medicine when we see that when we diagnose a patient on the basis of the various signs, or you say the symptoms. So what I wanted to say, that when we talk about this histology, the evaluation of histology, our understanding of this histology is related to the closely linked to the histopathology, we can say. An early work, you can see in this image over here, that is the image of the Ptolemy. So this early work of the Ptolemy described the refraction, the model of the refraction. So although this Ptolemy, he didn't recognize its use for the magnification, but later this Alhazen explained how this convex lens could produce the magnified images in his book that is called as the Book of Optics. So this idea was not widely applied for almost 200 years, even though this model of refraction or this magnification was discovered by this Alhazen. Then later on, what happens, that this Roger Bacon, during this Renaissance, he expanded on Alhazen's work, right? And he described the important optical principles in his book, which is called as the Opus Majus. So what he proposed, that the clinical use of magnification, which is likely influenced the later development of the spectacles, although the exact origin is still uncertain, not clear, but that was the proposal, by that this was the use, the clinical use of the magnification, and that later on develops as the spectacles. Now, later on, what happens, that this Roger Bacon, he described these optical principles in his Opus Majus, building on the Alhazen's work. Now, as I mentioned, that he proposed the clinical use of magnification. Later on, what happens, that many of the instruments from the period when the simple wooden tubes with a convex lens at one end and the flat glass plate will be there at the other end. So because they were often used to observe the small insects, we say, it was popularly called as the flea glasses. So later on, this flea glasses was discovered. Now, after that, what happens, that there was a French scientist, this French scientist who is called as the Petrus Borelius. This described in his book that is called as the Historiarum et Observationum Medico-Physicarum. So in this book, he described that the structures which are seen in the human blood, which are likely the erythrocytes or the clots, so these were represented by some of the earliest, you can say, the microscopic observations of the human cells and the tissues. So although his work, that is the Petrus Borelius, was later overshadowed by the different scientists, the other scientists, later on what happens, that in 1661, this Marcello Malpighi, he used the microscope to study the frog lungs, and he discovered the capillaries. Now, when this Marcello Malpighi, he discovered these capillaries, he provided the missing link, which was not there between the arteries and the veins, which were discovered by the William Harvey. Now, this showed that the blood circulates in a closed system rather than seeping through the various tissues, we can say. Later on, what happens, that this Malpighi, the Marcello Malpighi, his findings was mentioned in another text, that is the D. Pulmonibus, and he demonstrated the power of the microscopic in the study tissues. And because of his contribution, later on, he was called as the founder of the modern microscopic anatomy and the histology because of his contribution. Now, when we see over here, this Robert Hooke, he enhanced the, you say, the compound microscope. Now, this Robert Hooke, by using the 3-lens system, he allowed the greater magnification. What we seen earlier, the 2 lenses were there. One was the convex lens and another was the flat lens. So this Robert Hooke, he discovered, or he allowed the greater magnification by the 3-lens system. Now, in his landmark work, which was published in as the Micrographia in 1665, he described the structure of the cork and coined the term the cells, the Robert Hooke, and he gave the concept that became central to the histology. Now, meanwhile, what happens that this Leeuwenhoek, right, he refined the simple microscope by using the polished single lenses, and that achieved the superior magnification above what was discovered by the Robert Hooke. Now, later what happens that this Leeuwenhoek, he observed some of the bacteria or the blood cells, you say, and even the protozoa, and which was later on referred as the animalcules. Now, how this rise of histology was taken care. Now, when we, as we discussed, that the microscopy and the lens making that made the great progress. But when we talk about the early 19th century, in the early 19th century, the histology was not yet widely accepted in the medicine. But at that time, the microscopes were often used for the practical tasks, such as you say to identify some of the bacteria, you say, or better exactly it is called as the Trichina spiralis, which was found in the meat. So that was used for the clinical diagnosis. Now later on, what happens that this Peter Muller, he emphasized the role of the microscopy and in understanding the various kinds of the disease. So in his work in 1838, what he explained that the disease involves the changes in the normal tissue in its structure, or you say in the function. So this became the basis of the histology and the histopathology in the clinical practice, or you say the medical practice. So when we talk about this Peter Muller, his students were there, like we say the Rudolf Virchow and then Schwann, and apart from that, one of his student, that was the Henle. So they played a major role in shaping the modern medical education. We'll see in the coming slide over here that this Henle, whose full name was the Jacob Henle, he was one of the student of the, as I said, that this Johannes Peter Miller, 3 students were there. This Jacob Henle, his approach was that he classified the epithelial cells. You can see over here the columnar cells, the ciliated or the squamous epithelium. And in his work that is widely known as Allogemny anatomy, right, he emphasized that the body structure and function are based on the cells and the tissues. And this marked an important step in the, you say, the emergence of the histology as a branch of anatomy, we say. Now, when we see over here, as the microscopy advanced, what happens that slowly it shifted from the quality of the tissue sample, right? Rather than focusing on the microscope, it was shifted to the quality of the tissue sample. Early sections were poorly fixed and it was quite thick. Now what happens that to overcome this problem, which was poorly fixed or the sections were thick, there was another scientist whose name was this Benedict Stilling, or we say the Wims. His work was continuously to improve this, and that led to the development of the modern microtome. Now, this enabled the thin, precise sections for improved microscopic analysis because of the discovery of this modern microtome. So the scientist, whatever the thinner sections they used to get because of the discovery of this modern microtome. So that over here, what we wanted to say that there was also issue in the preservation of those tissue, the fixing of those tissue. So even though the microscopes or you say the section cutting methods were greatly improved, but the tissue would quickly decompose or putrefied. So there was a major breakthrough by this Alexander Butler, who discovered this formaldehyde. And because of the discovery of the formaldehyde, in 1858, what happens that the prepared solution was formed, and because of that, the tissues were fixed properly. So this Ferdinand Blum identified this formaldehyde as a tissue fixative in 1893. And those time, the 4% solution was commonly used. This was originated from the work that was by the Ferdinand Blums, which was used initially. So what I wanted to say over here, that by now we had this modern microtome, and even the fixative agents were discovered that was used as the 4% formaldehyde solutions. Now, when we go ahead, the tissue staining was completely changed. And that was because of the, we say, the unstained tissue section, which was almost transparent. And because it was transparent, it was very difficult to see the details in the clear way. And because of that, when we see over here, one of the most important stains, that was hematoxylin. All of you know that it is a purple basic dye, and which was obtained from the logwood tree. So because of the discovery of this hematoxylin, it was first used by the Mayans as a fabric dye and was later brought to the Europe by the Spanish explorers. Now, what happens in the 19th century, this Waldeyer tried using it in histology, but it was really successful in 1865 when this Bomber used this oxidized hematine with alum as a mordant. Now, second important this dye was discovered, that is the eosine, which all of you know that it is a pink acidic dye, and it was introduced in 1875. So this eosine, it stains the cytoplasm, as all of you aware, especially the hemoglobin rich areas, but it doesn't stain the nucleus well. So it was used as a counter stain to the hematoxylin. Now what happens that together this hematoxylin and eosine became the most widely used staining method in histology, not only histology, even in the histopathology. Now, that continues that we all know the hematoxylin and eosine technique, but just to give a brief idea, this hematoxylin eosine is the standard stain for demonstrating the nucleus and the cytoplasm. When we see this hematoxylin, it acts as a basic dye and used with the alum, and basically it stains with the nuclei, and the color, the outcome will be the blue or the purple, and the cytoplasm will be the pink, as I mentioned previously, that the another stain, that is the eosine, which the cytoplasm stains, and it is an acidic dye, right? So these two played a vital role in the staining. Now, apart from this hematoxylin and the eosine stains, there were some special stains used as well. And because of these special stains, there were some limitations of the hematoxylin and eosine, and because of that, the presence of this special stains, which was highlighting the specific tissue components. Now, this enabled the identification of the carbohydrates, the lipids, proteins, and you say the connective tissue fibers. Apart from that, it also improved the visualization of the microorganisms like bacteria or the fungi, you say. Further, it facilitated the differentiation of the normal versus the pathological tissues. Apart from that, it also served as the foundation for the, you say, the immunohistochemistry or the molecular pathology, which is widely used in the research purposes nowadays, also in the histology, or you say the anatomical research as well. Apart from that, it was also supported by the development of some staining techniques like you say the pass staining, or you say the Mason's trichome or the silver stain. So there are some special stains that also added in the staining process. So as I was mentioning about this immunohistochemistry, this technique changed the histology from just looking at the tissue shape under the microscope to a more precise, you can say the molecular, or you say the functional form of diagnosis. And because of this, it is also called as the Brown revolution in the histopathological sciences, you say. Coming to the further development, that is regarding this optics to the algorithms, with the arrival of the computers, as soon as the computers arrived, it was only a matter of times, we say, that this microscopy and the digital imaging also came together along with the computers. And because of the when this computer came, this microscopy and the digital imaging came, this allowed the histological images which can be stored, or you can say that it can be shared and it can studied in a much better ways. So the initial system, that is the at first we say that the systems were using this motorized microscopes and the cameras to capture the various still images. Now it has been joined together to make you say the digital montages will be there, and because of the files were very large, whatever the digitalization of the slides were there, it was difficult in the beginning. Later what happens that by the end of you say the 1990s, the whole study whole slide imaging came, and because of the whole slide imaging, the transformed analysis, storage, and the retrieval of data became very easy. So that was because of the growth of the World Wide Web, you say, the digital histology expanded further, and because of that, even the global collaboration is very well promoted and done, and that is enhancing and enabling the better education of the histology, we can say. Then, as I was mentioning about this mode of digitalization, you can see over here, this is the slide digitalizer. So there will be a glass slide which is scanned using the slide scanner, and that produces a high resolution digital image of the tissues. Then with the help of the image cutter, the large image is divided into the small tiles, we say, and that helps in the storage and viewing the faster, and you can say the more efficient. Now what happens that these data, right, this in the metadata, it stores the information about all the slides, and this helps in organizing and retrieving the images. Apart from that, whoever is the viewer or the observers are there, they can mark the important regions like the different cells, you say, or the abnormal cells. So annotations was there, and that was helping in studying the slides to the various students across the world, you say, sitting in different areas in a better way, right? And then apart from that, there were some tools which help the pathologist in viewing and annotate also and analyzing the different slides. So this was helping in the not only the students but also the researchers or the clinicians also. Now then came the era of the virtual microscopy. Now this virtual microscopy, when we say, it was possible to add annotations. You can see over here, you can mark the different cells and different structures over here, and that will help the students sitting at different corner. They can go through and individually, without the help of the instructors, they can identify those things. So this was the biggest benefits, you can say, and millions, or you can say the hundreds of the students can look and get the ideas of the different structures. Now, because of this, you can say when you compare this traditional versus this modern technology, so histology can be difficult because it involves the many various different organs, you can say, and that includes the 2D sections, right? And so identifying the structures is not always easy in the 2D sections. So we have to apply our brain, the mental reconstructions we do to analyze accordingly the 3D structures from the flat images. And that becomes even harder when many tissues will be looking the similar with the usual, you say the hematoxylin stain, that is the pink and the purple color. So it is really difficult to see in the 2D images, right? So because of this, we can say that the histology learning depends on the visual literacy, and that allowed us to recognize the patterns and the details very quickly because of the technology. So in traditional labs, when we see the students uses the microscopes and they will have 100 to 300 slides. Whereas when we talk about in the digital era, the technology, you can have n number of slides from the different sources that is there, and this will help in the, you say, the curriculum defining or the changing, and that will transform the way histology is taught. So these are the different aspects of the traditional, or you can say the modern technologies. Now, when we see the concerns about this virtual microscopy, these are definitely the nostalgia will be there, traditional bias. So these comes about the when it comes with the virtual microscopy, the many teachers, we say, or the students, they resisted initially because they were strongly attached to the traditional training methods, and they were worried about the reducing the hands-on skills, because when they thought that if we see this virtually, right, the hands-on training will be not be there, or it will be reduced. So another concern was the virtual slides, which were often too perfect, because whatever the images were taken was taken of the best slides. So, and when we do in the traditional methods, there will be all kinds of slides. So they will have better exposure. So that will reduce the curiosity. And even because of the perfection of the slides in the virtual microscopy, that reduces the motivation, because then you don't need to explore the things in the different ways, right? So these are the different concerns about the virtual microscopy, that the perfect slides, the digital distractions are also there, and there will be the loss of practical skills will be there. So these are certain concerns about the virtual microscopy. Now, when we talk about the counter arguments, the main counter argument is that the virtual microscopy still needs the active learning. Why it is so? Because students still have to navigate the slide and identify the particular region, the right region. So the process is not passive like simply looking at the pictures. Apart from that, we can say that various studies has also been conducted that the learning with the virtual microscopy is equal to the traditional microscopy as well, right? And in some cases, the studies says or shows that this virtual microscopy is better than the traditional microscopy. And another evidence I can say over here, that this FDA has approved in 2017 that whole slide imaging, which supports this reliability for the diagnosis also, not only about the teaching and learning. So apart from that, what I would say that the evidence suggests that this virtual microscopy is not just a replacement of the traditional microscopy, but it can perform equally and even sometimes better than the traditional microscopy. So definitely there are some limitations of the virtual microscopy, like the high costs of the scanners, the software, the storage, the IT support in so many medical institutions. Still, there is a lack of the infrastructures. Even sharing is difficult because open access will increase the server loads and the costs also. And then there will be the limited virtual slides, maybe 100 to 500, and the reduced exposure also to tissue variability. So these are the certain limitations of the virtual microscopy. Definitely, when we talk about the use of the social media, it can enhance, but every students are nowadays using social media, but basically they are not using for the educational purposes. That is the drawback. But when we talk about this virtual microscopy, the use of the social media, when we talk about the Twitter or the X, it can be used for the quick updates, or you can say the hashtags, or connecting with the experts, or you can say for the networking. Similarly, the Instagram, which is particularly used for the images, so it can be used for the histology, and it can be used for the perfect, you say, annotated images or the quiz posts also. So the social media can be used in an appropriate way, but unfortunately, the students are not using the way they should use for teaching and the learning methodology. Coming to the another, that is the virtual reality and the augmented reality. The virtual reality and the augmented reality have been used in this anatomy education for over a decade, I can say now, but their application in histology is still limited. And early virtual microscopy systems were even described as a virtual reality, but I can say that the modern virtual reality typically uses hand-mounted displays, while this augmented reality is often delivered via the smartphones, or you say the tablets, right? So what I wanted to say that this can be used, but still there is a lot of works to be done. So users could navigate slides using the simple hand gestures for zooming and movement, you say the simulating a virtual microscope experience, but it is not widely used at present for this teaching and training of the histology. Apart from that, another we can discuss about this 3D printing. This 3D modeling in histology is a very recent development, or you can say it is emerging right now. So virtual epithelial models also it adds in the learning and the teaching methodology, and it is used basically for the online learning. And these models can also be adapted for the virtual reality. Apart from that, when we talk about this confocal macroscopy, it can produce the 3D models of the tissues and the organs, and that can be again used in the virtual reality based teachings. So these are the recent developments. Apart from that, already about this artificial intelligence applications has been discussed in the morning lectures. But when we see this this modern pathology, it is definitely becoming more digital, and slide, and we can use them for the diagnosis. And this is very important because the number of diseases is increasing, and even I can say that the funding is decreasing, and because of that the pathologists are work even when we talk about the workforce of the pathologists, it is getting older and smaller. So image analysis algorithms, it can automate or the semi automate tasks such as you can say the immunohistochemistry, or it can be used in the quantification. So just to conclude, I have 1 minute over here. So I would like to say that the histology, we can say that it has a long and the fascinating journey. We can say that from looking at a cells under a microscope to understand the disease in a deeper, or you can say in a more precise way, or at the cellular level, right? It remains the backbone of the diagnosis. You say not only in the diagnosis, but you say the grading, staging, or even in the treatment, or you can say especially the diseased conditions like cancer, it is playing a vital role. And with the help of, or you say the newer imaging, the molecular tools, we are now able to see the structures at much deeper level than what we used to see earlier. So I can say that the future diagnosis may rely even more on the digital and the molecular data, but still the histology, I can say that it will always need, or you say the scanners or the digital platforms to keep moving the forward. I can say in India, when we talk in our country, the traditional slide based learning is still very much alive in most of the medical colleges, but I would suggest that the virtual or the digital learning are still steadily growing. We can see in some of the institutions like AIMS Delhi, and in various institutions we say that the histology labs, they are digitalizing. So I would say that these will make learning more flexible, more connected, and more accessible for the students and even for the teachers all across the globe, I can say. So the future of histology, I would like to say that it is not just about the technology, but it is about using the technology in a proper way, wisely, to improve the science, the medical science, the teaching, or even the patient care, or in diagnosing the diseases. So this is all about from my side, the journey from the previous to the present era. Thank you. Thank you so much.
Ma'am, you are muted
Ma'am,
actually.
unmute your device, ma'am.
Sir, excellent presentation from your side, sir. From
Thank you so
journey
much,
from the
ma'am.
tradition to innovation, sir. Sir, I would like to ask, sir, at present in your institution, what are you practicing, sir, for this microscope, please?
Right now, ma'am, here we are working on the traditional mode only. But
Oh.
when we were talking about the previous institutions, there we have digitalized. Here we are just following the traditional ways,
Okay,
right?
sir.
Not we have not digitalized, but we are in the process of digitalizing the histology lab. We have developed the dissection hall, the museum. As I joined last year, only lab I need to develop now is the histology lab, and I have a vision that by 6 months, next 6 months, our histology lab will be completely digitalized, because what I feel that each student should have an access of the computer, and whatever we focus, right, during the demonstration of the slides, the each student should be able to see what we are focusing, because when the students they see the slides in under the microscope, they will be struggling, because only the students they are knowing that what they are seeing, right? And it will be very difficult, because the number of students are increasing in each and every medical college. 250 students are there, right?
Oh,
Individually, you
okay.
cannot go to each and every students and focus their area in the slide and guide them. So it is very necessary that we should digitalize this our histology lab, so that with the pointer we can focus and every students can see that what we are focusing and what they need to see in the particular area. So it is I say that it is the time that all the histology lab should be digitalized, but we should also use the traditional method, but digitalization is mandatory. I feel I recommend that, sir, ma'am.
Okay. Sir, actually, sir, in the early 2000s, sir, one of our dean was very kind enough to supply us the microphotography. So the old
Right.
one with the camera, and we utilized that microphotography. And once when we finished the classes, we used to project it in the microphotography, and we used to teach them. I think Dr. Sulachna also was there when I hope you remember Sulachna when you were here.
Yes,
You
ma'am.
were
Yes,
teaching
ma'am.
the students with the microphotography.
Yes, ma'am.
That was a very primitive. That was one when the technology was evolving, sir. So I did government setup. I fought with the dean, and I got it, and we were able to do it, sir. And now also we are a newer medical colleges are having computer assisted video assisted recently they have given for all the 11 new medical colleges. So I am also trying to get for that also. And your presentations are about the students digital distraction. That is the main problem here, sir. And the loss of practical skills, that is one thing which is
Very,
growing,
very,
sir.
very
Yes, sir.
big issue, ma'am. Even in the
Yes, sir,
lecture
sir.
theaters, they
Yes,
will
sir.
be using all the gadgets,
Yes,
right?
sir.
So, and because of that only, because nowadays you see that the students are referring to the textbooks. They will
Yeah,
be
yes, sir.
going through the YouTubes,
Yes,
right?
sir.
But those YouTube contents are not
Yes, sir.
peer reviewed, right? And when you ask questions, right, that which textbook you have referred, they will not be mentioning
Sir,
the
here
textbook. They will
nowadays
see.
if we ask any question immediately, chat GPT, sir.
Yes,
That
Chat
is the
GPT,
problem here.
Perplexity, so many Google, so
Yes,
many
sir.
things are there, ma'am.
Sir, once
Right?
I put forth one question, immediately all the 100 students gave a right answer, the same answer. Then only it struck my mind that they are going for this chat GPT. So that
Right.
was a very good
That
presentation,
that
sir.
way they are
Thank you so much,
misusing,
sir, for
ma'am.
it. Yes,
But
sir,
one
they are
thing I would like
misusing.
to
Thank
add
you,
what
sir.
you asked the previously, ma'am, that
Sir.
the many of the medical colleges now they are trying to develop their labs. And what you mentioned that each and every histology lab or the research labs, they are having the pentahead microscope. Most of the institutions I see that those like pentahead microscopes, if they are having, it is very easy to teach the postgraduate students with the help of
Oh,
those. Even
yes.
the some of the microscopes they are equipped with the cameras and all, so that they can click that particular
Sir.
area and then they can display to the students on the you say the
Histology they fight and get it, sir. But for anatomy, it's every time a question mark for us. And sir, one more thing, sir. You told about that digitalization, that www.researchgate free. Is it a free access or we have to pay for that, sir? that
Pardon, ma'am?
that digitalization, where you are saying that free access, no, sir. Can we
There,
go to ResearchGate?
ResearchGate, so you can access, ma'am. But
Oh,
there are like some institutions are there. There you will be able to see the slides. They have
Okay, sir.
already, it is available and
Okay, sir.
freely available. You can access, ma'am.
Oh,
The
okay,
astrology
okay,
slides.
sir. So thank you so much for clearing our doubts and very well explained and a remarkable presentation. Thank you once again, sir. And
Thank
over
you,
to
ma'am.
Slochana for the. Sorry, I've taken much of your time.
Thank you, sir. It was a fascinating journey, sir, from early days to light microscope to virtual microscope and now to the artificial intelligence. That was a lovely talk, sir, informative talk. Thank you so
Thank
much,
you.
sir.
Thank you, ma'am.
Thank you, Dr. Rajan.
Sir, good afternoon, sir. Dr. Rajan here.
Thank you so much, sir, for giving me an opportunity.
Thank you
And
so much, sir, for joining this conference and giving a visibility towards all anatomists. Thank you so much, sir.
I'm really grateful, sir. Every time you invite me, and I feel honored to be part of this conference. And I feel this is the only anatomy conference which is regularly being conducted every year almost.
Yeah, yes, sir. Yes,
So
yes.
it's a really great thing because many days nowadays I see that people are not able to go physically, but at least they can attend these virtual conference, e-conference, and they can present their paper. So that is a very good thing, sir, and I appreciate that way because nowadays for the PGs, it is mandatory to present the papers. There are chances that they are not able to go and attend the various conferences in person. So at least these are the platforms where they can present their works. So I appreciate your efforts, sir, in that.
Thank you, sir.
Thank you, sir.
Ma'am. Thank you so much, ma'am.
Sir,
Thank
Dr.
Thank
Rajan
you,
is
Dr.
very,
Rajan.
very unassuming. It's a very quiet person, Dr. Rajan.
Thank you.
Ma'am, Sulochana, ma'am, can you see any queries in the chat box, ma'am?
No, Dr. Soumya. There are no.
No, nothing in the chat box.
Okay,
Nothing.
since there is no queries in the chat box, we conclude the session, ma'am.
Thank
On
you,
behalf
thank you
of
so much.
organizing team, we would like to honor a resource person with a virtual e-certificate and e-memento. Thank you, sir. We will also extend our gratitude to our chairperson. First, Dr. Sivahami, ma'am. Thank you, ma'am. Thank you for joining the session, ma'am.
Thank you.
Next, we honor our Sulochana ma'am with the e-certificate and e-memento. Thank you, ma'am.
Thank you, Doctor. Thank you, Dr. Soumya.
Thank you, ma'am.
Thank you so much. Thank you so much.
Thank you, Soumya ma'am, for conducting a wonderful scientific session. Let's proceed with the last scientific talk of the day. I now request Dr. Ravi Shankar sir, Assistant Professor, Department of Physiology, to conduct the session. Over to you, sir.
Thank you. Am I audible?
Yes, sir.
Okay. Good afternoon, everyone. On behalf of the organizing committee of BMS Econ 2026, I welcome you all for the next scientific session on brain mapping and stimulation techniques by Dr. Rohit Verma, Professor in Psychiatry, AIIMS, New Delhi. I'm Dr. Ravi Shankar, Assistant Professor in the Department of Physiology, Arba Medical College. I'll be moderating this session. So I would like to I'm happy to introduce the chairperson for today's session, Dr. Yogesh Kumar, professor in the Department of Physiology, AIIMS Patna. Sir, I'm actually privileged to welcome sir because I was working under him prior joining here. So, sir is a distinct professor of physiology with over 18 years of academic experience and 10 years of administrative leadership. The sir has he has joined as assistant professor in AIIMS Patna, and then he has progressed to professor. He has extensive teaching experience and research experience, as well postgraduate guidance and also DM guidance. Sir has served as a deputy medical superintendent for 8 years and a nodal officer for PMJAY Ayushman Bharat Health Scheme. Sir has a extensive research interest in clinical physiology, intraoperative neuromonitoring, heart rate variability, artificial intelligence in healthcare. Sir has authored over 60 publications and successfully completed 10 funded research projects. Sir is a recipient of 5 prestigious national awards, including Bharat Neuro Ratna Award for Excellence in Neurophysiology and Dr. Sarvapalli Radhakrishnan Best Teacher Award in 2025. Sir, are you there? Over to you, sir. I would like to give it to Dr. Yogesh, sir.
Okay, thank you, Ravi, for introducing me. And it is an honor and privilege to be part of this wonderful conference because I can see amalgamation of physiologist, anatomist, and biochemist here. So it is a great opportunity. And as Ravi told, he was here. We miss you, Ravi, here. You were a great help till you were here. I was not bothered much about my IONM services now. Every time we are taking care of IONM cases, I personally have to go there and we are waiting still for your replacement. So thank you. With this, let me introduce our speaker for today's session, Dr. Rohit Barma. Can you project his profile or should I
Yes,
share
sir.
my screen?
Sir, it is projected, sir. It
Okay,
is
okay.
already
Now it
projected.
is there. So, Dr. Rohit Barma is a professor of psychiatry at prestigious institute that is AIMS New Delhi, and he holds an unique and distinguished role as faculty in charge of both MECT facility and brain mapping lab at AIMS New Delhi. This makes him one of the most hands-on clinical expert in this field in the country, and his academic output is also very remarkable. As projected on the screen, he has been recipient of various awards. To name a few, he has received ICMR MD thesis award and received BB Sethi award and GC Boral award in 2014, and he has been honored with JK Trivedi and Valiant Award by IASP in 2022. So we are waiting to hear from you, sir. And he will be taking us through brain mapping and stimulation techniques. So over to you, Dr. Barma. And we all will be listening to you soon.
Thanks a lot, sir, for the kind words. Is my screen visible?
Yes, sir.
Yes, sir. Yes,
As
sir.
a,
It's
as a
visible.
whole screen, it's visible, right?
Yes, sir.
Thank you. First and foremost, I thank Dr. Gopinath and the team for having me here. As the previous chairperson just said, that I'm the odd one out here in this league of extraordinary gentlemen from anatomy, physiology, and biochemistry and others. I'm a psychiatrist, which has to do with all of your branches, but extrapolate and transformate your knowledge into our clinical senses. So I'll try to go through what has been happening in psychiatric perspective of anatomy and physiology, how we are dealing with the illnesses of brain that we understand from a different perspective nowadays. So the brain is what we are all concerned with here. It's an amazing organ, and it's it's a network of crisscrossing fibers here and there, which we consider as neurons. Neurons, which are the basic primary unit of this whole amazing organ, which has its multiple organ parts from a cell and an axon and dendrites and so forth. Initially, we have been thinking about the entire brain since the early 20th century as in a form of an anatomical structure only. So we divided into frontal, parietal, temporal, and then sulci, gyri,
And then we labeled each of these different areas as serving some of the other functions. All of this work was more or less governed by the homotypical architecture of the brain, which was what Brodmann and later on Penfield also thought about. That there are 6 layers in the brain, and each part of these layers subserves a different kind of function. But later on, it was found that not all the brain areas have this homotypical 6 layer structure, but there are heterotypical in structural senses, and different regions of the brain, they have different height, width of these different layers depending upon what kind of a function they are actually attributed to. And this is what has led to functional neuroanatomy. Let's just go through what has happened in the past few decades. The topmost conditions which are causing disability worldwide, and among them the top three, one of the top threes are from mental and neurological states, including depression, anxiety, strokes, and dementias. And how do we manage this? We manage them by medications. But if you look at the pharmaceutical market, the time to develop a drug is maximum for CNS drugs. The time for approval of a drug is least for a CNS drug, and the development cost is maximum for a CNS drug. All these things makes us a little bit of nervous that whether these agents that we are dealing with in a pharmaceutical manner, are they really capitalizing the states of mind and brain that we are addressing to. And with this comes forth this area of research, which is brain imaging. So what we initially understood in form of a structural anatomical form of structural brain imaging in form of computed tomographies and MRIs, magnetic resonance imaging scans. Gradually over the past few decades, we have now understood that the brain is much more than this. What we grossly see in a CT and MRI just gives us a, a very gross picture of the brain. What is happening underneath the neurons, underneath those layers, is not available through these structural imaging forms. So therein comes these functional brain imaging patterns. So in functional brain imaging patterns, there is something called as a functional fMRI. There is something called as diffusion tensor imaging, magnetic resonance spectroscopy. Then there is optical form of brain imaging called as functional near infrared spectroscopy. There is an electrical form of imaging called quantitative electroencephalograms and magnetoencephalograms. And then there are molecular imaging in form of positron emission tomography and single photon emission computed tomographies. Mind you, none of these are as of now able to diagnose an individual patient with any form of neurological or psychiatric illness, but they are all useful for research level activity and are there to help us to understand the functional neuroanatomy of an individual. So fMRI sees for different altered metabolic states and different cortical regions. It's just the simple MRI machines, but beyond the MRI, it gives us a bold response, the blood oxygen level dependent response, in which we get to see the different regions of the brain, how they are interacting. So what was seen through an MRI, that one frontal cortex is related to temporal cortex, but very different from the occipital cortex. Now in a functional MRI scan, two different areas, very far apart from each other, can light up simultaneously. And that tells us that those two regions are connected in some other manner, which is not grossly visible through an MRI or a CT scans. The MRS scans, magnetic resonance spectroscopy scans, they talk about altered levels of neurochemicals like choline, glutamate, glycines, creatinine, myosin. All of them gives us an understanding of about the neural architecture of the brain. And the diffusion tensor imaging scans, they give us how the tracts in the brain, particularly the axonal tracts, how they are placed and are connecting one region of the brain to the another region. FNIARS is one of the most recent advances in optical imaging. It is functional near infrared spectroscopy. It is akin to an fMRI, but unlike an fMRI, which can penetrate to the deeper labs of like an MRI, it can only penetrate to a depth of 2 to 4 centimeters. So it is only as of now uh, about to answer questions which are of cortical nature. So they can also talk about activated states of brain or resting states of brain. And the work in this is going on from the past few decades. Electrical imaging in form of electroencephalograms have been there from now nearly 90 years. And we have seen a lot of waves in our brains which are present not just during the daytime but also during night times. The pattern changes, of course. We have more of beta alpha activities in the daytime and more of delta theta activities in the night times. And the function of the brain depends upon how these waves interact with each other. And machines based on these interactive wave patterns are now coming up, which talks about how do these wave patterns result into certain form of a mental state or a neurological form. These electroencephalograms, when they are quantitatively analyzed, they can give rise to something called as a waveform or a topography or a source localization in form of LORITA or low optical resolution electrotomography. The waveforms are in form of event related potentials. Prior to few decades back, we labeled them as evoked potentials, but we found out that the brain remembered prior to a stimulus what was going to happen in form of waveforms which were happening just prior to a stimulus happening in form of readiness potentials, Berishka potentials, or contingent negative variations. And that's why we changed the name from evoked potentials to event related potentials, that they can happen before and after the stimuli. So after the stimuli, there are early waves which are mostly sensory. There are mid latency waves, and then there are later event potentials which are mostly related to one of the other mental states of the brain. Then EEG can also give you a form of a cortical source densities. These are also important for epileptic patients and also utilized for some of the other space occupying lesions when we are trying to find something of a sort in the brain from the scalp itself, what kind of a network connectivity one region of the brain is having with another. Just like in EEG, there is something called as an MEG, magnetoencephalograms. Magnetoencephalograms are a fusion of MRI and EEG, in which you are able to go through a full depth like an MRI and are able to see the electrical activity of the brain. Since electrical activity and magnetic activity are perpendicular, they are both one and the same thing. There is nothing solely electrical, everything is electromagnetic. So these magnetic waves are another way of capturing the activity of brain through these machines, which can map the entire cortex, provide you kind of provide the entire uh, area of the brain without providing any kind of distortions while passing through the skulls. They also give results in form of, just like in EEG, in form of waveforms, in form of cortical source densities, and in form of localized areas of deficits where the dipoles are altered. Molecular imaging in form of selective and non-selective molecular imaging exists through PET and SPECT. The ligand-gated molecular imaging radiochemistry are selective ones, which can give you activity about a particular specific receptors. Like for example, in psychiatry, the occupancy for dopaminergic D2 receptor tells us about how much the dose of a drug is required for 65% binding to this receptor, which will produce the effect without producing a side effect. So these kind of scans are helpful in defining those kind of parameters. Another diagnostic form of these kind of scans are in form of a TRODAD scan, in which Parkinson's patients are diagnosed with TRODAD scan. And nowadays with F-DOPA PET, they are also delineated than other Parkinsonian states like MSA or PSP or drug-induced Parkinson's. Non-selective molecular imaging like SPECT and PET can also talk about mapping the blood flow or the glucose metabolic rates in different brain regions. They're also useful in many regions, many scenarios, like for example, depression. We often find hyperfrontality in the cerebral blood flow and glucose metabolism, which differentiate them from healthy patients. All these kind of brain mapping techniques, they tell us about and teach us about a paradigm shift, that not simple neurochemical alterations are responsible for brain altered or uh, brain dysfunctions, but a circuitry based phenomena may also be important to understand how the brain really functions. And so we have come to a very fine conclusion at the end, that electricity is the currency of the brain. And it's the entire axonal length where all this electricity works. And at the synaptic level, the chemical part happens. So medications, they work at the synaptic levels, and brain stimulation techniques or neuromodulation techniques, they work at its axonal levels. And so we are gradually shifting from the neuropsychopharmacology kind of states, which were dependent upon neurotransmitters, to a circuitry based neurotherapeutics in form of neuromodulation strategies. All of these are based upon the various kinds of current which are flowing through the transmembrane of the axon, and are delivering at the end at the synaptic cleft in form of EPSPs and IPSPs, which give rise to action potentials at axon hillock, and then give rise to vesicle fusion at the synaptic cleft, leading to again to the conversion of chemicals from one presynaptic cleft presynaptic neuron to the postsynaptic neuron, leading to the next generation wave of electrical currents. And these electrical currents in the next postsynaptic neurons are maintained, the effect are maintained from this noble concept of long-term potentiation and long-term depression, mediated through glutaminergic NMDA and AMPA receptor profiles. So nowadays, lot of stimulated techniques, brain stimulation techniques are available, which can address these kind of circuitry based neurotherapeutics. There are techniques based on direct electrical waves. There are techniques based on magnetic waves, optogenetic, thermal, acoustic, mechanical, and chemical. All sort of energy kind of mechanisms. I won't go through all of them because there are too many of such techniques available nowadays. I'll give you the gross of what are the three most commonest one that we are using as of now to change the brain states. So this is the plethora of what the current brain stimulation techniques look like. They range from non-focal to focal and from non-invasive to invasive. We have the most invasive in form of deep brain stimulation and vagal nerve stimulation. Vagal nerve stimulation also now have a non-invasive form in form of transcutaneous vagal nerve stimulation. Optogenetics is right now only majorly in the realm of animal models. It is highly invasive. Then for the convulsive or caesarogenic form, which somewhere lies between non-invasive and invasive, is magnetic seizure therapy and electroconvulsive therapy. Completely non-invasive are transcranial direct current stimulation, repeated transcranial magnetic stimulation, and focal ultrasound techniques. These are the most commonest ones which are currently available. There are many others like photobiomodulation, TAVS, TRNS, and others which are currently in research practice. They have not yet reached the clinical market, but these are the available machines and techniques which are available in the clinical market as of now. MECT is modified electroconvulsive therapy. Electroconvulsive therapy began since 1930s, in which it was found that generating a seizure was leading to changes in the brain state, particularly in form of improvement for depressed as well as schizophrenic or catatonic individuals. And gradually over the past century now nearly, we have developed this machine from ECT to MECT techniques, utilizing anesthesia, utilizing muscle relaxants, changing the technique in form of lower current strands, changing the charge, developing constant current machines, having an EEG monitoring status. All these have reduced the lot amount of cognitive side effects which are initially present with such machines.
Dr. Burma, I think your voice
MCT to
is
a patient.
yeah.
RTMS or repeated transcranial magnetic stimulation is another second modality which is currently very much in use nowadays. It was started since 1980s by Dr. Barker. It works on the similar principles that electricity and magnetic fields are related. And when you flow electricity in a loop, the changing electric field will generate a magnetic field. That magnetic field can penetrate into the brain since magnetic fields are not stopped by skull or tissue. They penetrate into the brain at a depth, and therein, since they interact with neurons which already are electrical in nature, they can generate electrical currents in them, just like an ECT. So what happens is a machine develops, delivers magnetic flux over the superficial cortical layers, from where the impulse in form of electrical nature goes to the deeper cortical layer, and from the deeper cortical layers the neurons again back propagate and reactivate the superficial cortical layers. So all these lead to stimulation of the dorsolateral prefrontal cortex and anterior cingulate cortex. These are in scenarios for depression, and they bring about change and improvement in target symptoms. And this is where we have found use of TMS providing benefit in depressed individuals. So many different areas of the brain are stimulated depending upon what we are trying to instigate. Currently, there are protocols which were initially or 30 to 40 minutes, had more than high number of sessions, were very costly and required many visits to a hospital. But gradually over the years, now we're making the modulation parameters modulating and making it better, faster, as well as cheaper. While the activation at one point can spread to an entire brain, the shape of the coil prevents that. So something called as a butterfly or a H-shaped coil makes the stimulation much more focal. A conical coil makes it focal to a deeper strength. Normal depth to which a TMS can stimulate is about somewhere around 2 centimeters. But nowadays we have something called as an H coil, which can go up to the strength up to length of 4 to 6 centimeters or even much more deeper, but at the cost of losing its focality to a large extent. There are varieties of TMS in form of low frequency, high frequency, and theta burst. Low frequencies are when you deliver one pulse per second or less. High frequencies are usually attributed as 10 pulses per second or more. Theta bursts are based on principles of theta gamma fusion, which happens usually at the night when we are all asleep and the memory consolidates. So they deliver theta form of pulses in gamma ranges. So delivering nearly about 600 to 800 pulses in IT base or CT base kind of intermittent or continuous theta burst stimulation. All in all, low frequency RTMS or low frequency RTMS leads to inhibition of the cortex. High frequency RTMS leads to excitation of the cortex, and ITBS or intermittent theta burst leads to excitation of the cortex, and CTBS leads to inhibition of the cortex. So when we want depressed individuals to become better, and we find that on the PET spec or FNIRS, the activity of the dorsolateral prefrontal cortex of the dominant hemisphere is lower, we stimulate that particular area with an high frequency RTMS or ITBS and make them better. And over the years, we have found that these kind of IT-based protocols, which were initially for long period of 37 minutes or so, when we changed them to theta burst protocols, we have reduced them to 3 minutes per session for So there are a lot of work happening with TMS, making the treatment come as closer to reality as much as possible. Now, all the brains are not always same. That is one of the most problematic scenarios for science. Population studies in different cultural phenomena, Chinese, Malay, African Americans, Europeans, Indians even, we have seen that the morphology of the brain is different. And for its capitulation, there's something called as neuronavigation, which is happening. In neuronavigation, someone takes an MRI of an individual patient, then co-registers it at a functional or a structural imaging scan, and then map that structural scan on with the functional activity, and then directly stimulate that functional region with a TMS or any kind of a stimulatory modality. Another problem is head movement, in particularly in TMS. In ECT, the patient is anesthetized, so they do not move a lot. But in TMS, they are not anesthetized; they are sitting just lying down on a small chair. So there can be slight movement, and since you are trying to deal here with millimeter kind of an area, even slight movement of the head, this is too much, but even slight movement of the head will lead to fall of the particular area to a different localized region. So for that, there is something called the robotic TMS coil available, which moves along with the head, compensating for the activity there. And it precisely maps the cortex and delivers the stimulation where we are actually intending to. There are a lot of protocols which are approved and a lot of conditions for which TMS has been now approved, ranging from depression, OCD, anxiety, smoking cessation, migraine, and so forth. Currently, there is something called as personalized TMS, which is ongoing, in which, depending on an individual's brain activity in form of EEG, there can be online or offline TMS provided to such individuals. This is our lab at AIMS. We have a new lab now, so this is an older photo, but we can see we have this robotic coil as well as a neural navigation approach for providing stimulation to a patient. The third one, which is commonest nowadays, is transcranial direct current stimulation. TDCS is one of the most simplest of neuromodulatory techniques. It is as small as a mobile device, even smaller. And there is nothing other than just two wires coming from it, which stimulate two different regions of the brain, one cathode and one anode. And the patient remains conscious all throughout the procedure. Akin to RTMS, an anodal TDCS stimulates the brain, and an anode TDCS stimulates the cathode. Plug, the action potential arises and the current flow inwards. For a cathode, the opposite happens, and that's why the brain becomes inhibited at the cathodal region. And this polarity effect is what is being utilized in TDCS. It doesn't generate an action potential like a RTMS or an ECT, but it makes the possibility of action potential happening easier. So in that sense, it modulates the neural cortical activity in form of inducing neuroplasticity. It doesn't cause excitosity, but it causes a continuous stimulation form. TDCS conventional form is in form of a anodal and a cathodal stimulation through two sponges. But now from past few years, we have something called as a high definition TDCS, in which there is a center anode or cathode surrounded by a ring of cathode or anode. So if you want to stimulate a brain at a very particular focal region, you can make that central region as anode and the surrounding cortex as cathode, so that the current will focally deliver only to that particular region and not dissipate somewhere else. And such a technique can be utilized to have much more precise stimulation at a particular focal region, just like a TMS. There are lots of varieties of TDCs nowadays available, and recently, last December, we have home-based TDCs approved in US FDA. There are various other forms of TDCs also available, called something called as a transcranial alternating current stimulation and transcranial random noise stimulation. These are variants which also utilize current, but not in a direct form, but in a form of a alternating wave, which utilizes temporal summation effects to deliver the neuromodulatory activities. TDC is one of the most simplest of the techniques. As you can see here, the patient is just lying there and there is a cap over his head on which the electrodes are placed. That is delivered for a period of about 20 minutes, on which duration he or she can relax, read, write, and nowadays we are asking them to involve some of the other cortical activities too. So with this, I'll come to an end with a take-home message that there are a lot of brain mapping and techniques available nowadays, and they have paved way to a lot of stimulation techniques also, which are all available as of now with the different levels of evidence. They are paving way in my field as an interventional psychiatrist, uh, which suggests that neuromodulation seems to be a promising approach. We have seen patients who become better without medications, without any therapy, just by neuromodulation. We have seen patients who become better after neuromodulation, even though they have not responded well enough to medicines. So, to me, within the safety limits of current approaches of this neuromodulation, future with these kinds of brain stimulation techniques appear bright. I thank you all for having listening to me, and thank you to the organizers and the chairperson for their time.
Thank you very much, Dr. Verma, for this very lucid and illuminating presentation. What you have presented today reflects a global shift from treating brain as a black box to now mapping it to its functional architecture and then treating it. And many techniques which you described today regarding the mapping, we as a physiologist also utilize some of those while doing intraoperative neuromonitoring. So my physiology colleagues will also get benefited from this. And it was a nice presentation, and we are learning so many stimulation techniques. It is a overlapping field. Sometimes we also are venturing into this TMS, and hopefully we will be taking your help also if needed. So thank you very much for this presentation. Over to the organizers to taking this session ahead. Ravi, you are
mute, sir. You're
unmute
mute.
yourself.
Sir, I have a few doubts. Can I ask some queries?
Sure.
Sir, what are the conditions that these things are used for apart from depression? Is it only used for depression?
Oh no, I just gave depression as a
Example,
commonest one because that's the easiest one to understand for everyone. It is one of the commonest disorder in psychiatry worldwide, not just psychiatry but worldwide medical disorders. So depression is one of the commonest one in which majority of the techniques have been used, but it is also used for hallucinations in schizophrenia, for tinnitus, which we use it here. It's used for OCD, obsessive compulsive disorders. It's used for traumatic brain injuries. Is one of the favorite areas where I work in, because for those individuals there is nothing else available in this world right now. So I treat them from past decade with these kind of brain techniques and have some wonderful results. Then there are migraine is one another area where these works. Smoking cessation is another area where these works. DBS, which I have not pondered into. DBS works well for epilepsies, and as for Parkinson's disease. Focal ultrasound works for essential tremors. There are work ongoing for dystonias in focal ultrasound as well as deep brain stimulation techniques. Uh, these are one of the most commonest one which are available as of now.
Thank you, sir. Is it combined with medications, all these neuromodulation techniques, or or is it used as a single therapy, sir?
Okay, so as I understand, science is a defensive branch. Whenever we ponder into a newer realm, we are supposed to first of all base our premise on do no harm. So for doing no harm, we are supposed to treat them with whatsoever best available evidence we have currently. So if there is a evidence of 70 years of medications, then the first choice that is provided to the patient is always medicines. Those who cannot tolerate medicines or who are not responding to medicines or who do not want medicines, for them we have this choice of providing an option of neuromodulation, and that is where they come in. Gradually, from the past 2 to 3 decades, I would say, initially all these, except for ECTs, all these neuromodulated techniques, they began with a single session only, just trying to delineate that they are not going to cause any harm. Now we have these accelerated protocols which try to treat individuals in a span of 5 days only. No medication can claim that. So the world is moving fast. We are trying to move fast, but within the confines of science and trying not to do any harm to any other individuals.
Thank you, sir. Thank you, sir. Thank you for your wonderful session. You took us through in a short span. You took us through all the imaging techniques and the neuromodulation techniques. Thank you for the wonderful session, sir.
Thank you for having
Thank,
me.
thank you, sir. Thank you for joining with us. And it was a very wonderful presentation also, sir. Having talking about the basic sciences involved in that, and very well you have talked about the neuromodulation techniques, TMS and DMS on which has a huge lot of clinical applications also. And I want to mention that, sir, definitely you are not a odd man out, and we are always this is an academic forum where we want to connect with the basic medical sciences and clinical implications, and that's where you being the right person to enlighten us on different clinical implications of different techniques also, sir. So thanks for joining us in spite of your busy schedule. And over to Dr. Ravi Shankar.
Thank you, sir.
Thank you, sir. Thank you, Dr. Yogesh, sir, for giving us the time and chairing the session. I would like you, since it's an e-conference, I would like you, on behalf of the organizing committee of BMS Econ, we would like to, as a token of our small appreciation, we are giving a e-certificate and e-memento to Dr. Rohit Verma, and then a token of appreciation to Dr. And as a token of appreciation to Dr. Yogesh sir for chairing the session, e-certificate and an e-memento. Thank you, sir. Thank you, Dr. Rohit Verma, and thank you, Dr. Yogesh Kumar, sir. Thank you, sir.
Thank you. Thank you.
I thank our speaker for the brainstorming session, and I also thank our chairperson and Dr. Ravi Shankar sir for conducting the session. As we conclude the scientific sessions of the day 1 of BMS Econ 2026, I thank all the speakers, chairpersons, moderators, and the participants for their enthusiasm and intellectual contributions. Let us continue to explore, engage, and innovate. Let us take a break for lunch now and proceed with the scientific presentations. I request all the participants to join the Zoom link shared to you to continue with the oral presentations. Thank you.
Hi everyone, we are closing this Zoom meeting now, and there is separate link for 2 p.m. meeting. All right? Thank you. Thank you very much.